Human physiology is the branch of science that deals with the study of human cells, the organs formed by these cells, and their organization to support the various functions of the body in health and disease. It focuses on the body's ability to maintain a steady state of homeostasis. Homeostasis is defined as a state of a stable internal environment maintained by living organisms through regulating various physiological processes while adjusting to external conditions. In this branch of science, human response to internal and external stimuli such as changing environmental conditions, changes in activity levels, and disease conditions is measured and is utilized for the betterment of human health. Levels of organization. The universe is made up of matter, defined as any substance that occupies space and has mass. Matter consists of one or more pure, unique constituents known as elements, i.e., hydrogen, oxygen, carbon, nitrogen, calcium, and iron. Atom is the smallest basic unit of these elements. Each atom is further divided into subatomic particles known as proton, electron, and neutron. Atoms combine to form molecules found in living organisms, such as proteins, sugars, and water molecules. Molecules go on to form the building blocks of the body's structure. The cell is the smallest, most basic, independent unit of life, which contains all the structures of the human anatomy along with different functions that take place in the human body. All the various functions are commenced by the cells. Each cell in the body is made of flexible membranes that surround the cytoplasm, a water-based, fluid-containing structure in which the operative units of cells called the organelles are suspended. Figure 7.1. Cells of similar structure and function group to form tissues and group of different tissues assemble to perform a similar function, forming an organ. Each organ is part of one or more physiological functions. A group of organs which perform similar functions make up the organ system that is important to carry out the physiological needs of the body. Figure 7.1. A typical human cell. A prototypical human cell comprises of internal organelles in the cytoplasm and inside the nucleus. The human body cell is made of extremely organized structures called as intracellular organelles, which contribute to cellular functions. Majority of organelles are protected by membranes consisting of lipids and proteins, which include the plasma membrane, nuclear membrane, lysosomal membrane, endoplasmic reticulum membrane, mitochondrial membrane, and Golgi apparatus membrane. Tissue types. Tissues are a group of cells with a similar structure that perform a specific function in the body. Tissues are classified into four basic types: epithelial tissue, connective tissue, muscle tissue, and nerve tissue. Figure 7.2. 1. Epithelial tissue, also known as the epithelium. It forms the covering of body surfaces, internal organs, and lines of body cavities. It also forms the lining of some passages and some glands. 2. Connective tissue. As the name suggests, the cells of connective tissue connect the cells and organs of the body together. Some of the important functions of connective tissue include support, protection, and integration of different parts of the body. 3. Muscle tissue. This tissue consists of cells specialized for contraction to bring about animal sciences. Movement. The cells of muscle tissue possess the ability to excite and respond to stimulus. 3 different types of muscle tissue are skeletal muscle, also known as voluntary, smooth muscle, and cardiac muscle present in the heart. 4. Nerve tissue. Found in the nervous system. Nerve tissue consists of specialized cells that can generate and transmit electrochemical signals in the form of nerve impulses as they possess an excitable characteristic that links different regions of the body and assists in the maintenance of homeostasis. Tissues are classified as epithelial tissue, forms covering the outermost surfaces of the body. Connective tissue binds cells and organs of the body. Muscle tissue excites. Figure 7.2. Different types of tissue. And responds to stimulus and contracts to provide movement. And nervous tissue connects between different parts of the body by propagation of electrochemical signals. 5. Epithelial tissue. Cells of epithelial tissue form the lining and covering of the outer surface of the body. The cells are organized with very little or no extracellular spaces present between them, attached to one another by means of desmosomes. The tight organization of cells renders epithelial tissue with the property of protection. They are anchored to a connective tissue which provides the cells with nourishment as the epithelium lacks blood vessels. The epithelial tissue is classified into two types. 1. Lining and covering epithelium. 2. Glandular epithelium. Lining and covering epithelium. As the name suggests, this class of tissue form the outer covering of skin, respiratory tract, digestive tract, reproductive tract, and urinary tract. Blood vessels, serous membranes, and other hollow internal organs are lined by epithelial tissue. The cells are organized in the form of a single layer or several layers. Lining and covering epithelium are further classified based on the shape and arrangement of cells into layers as big. 7.3. Simple squamous epithelium. The word simple refers to a single layer of cells that rest on the basal surface and give the appearance of thin scales. The cells are flattened, horizontal, and elliptical in shape with a centrally located nucleus. This epithelium assists in the exchange of nutrients, waste and gases, and protection. It is found in areas where diffusion or filtration occurs. Example: lining of the lymphatic system, alveoli of the lungs, and glomerular capsule. Simple squamous epithelium lining heart and blood vessels are known as endothelium. Similarly, simple squamous epithelium lining the outer surface layer of the serous membrane, such as peritoneum, is known as mesothelium. Simple cuboidal epithelium. Single layer of cuboidal shaped cells that have a centrally located round nucleus. This type of tissue is found in areas where absorption or secretion occurs. Example: forms the lining of kidney tubules, surface of ovaries, ducts of pancreas and thyroid gland, and anterior surface of capsule of lens of the eye. Simple columnar epithelium. Single layer of elongated cells that are column-like with a nucleus located in the basal end of the cells mainly for the purpose of protection, secretion, and absorption. Example: lining of small intestine and uterus. Simple columnar epithelium can further be classified into ciliated and non-ciliated simple columnar epithelium. Ciliated columnar epithelium has additional finger-like projections called microvilli on their apical surfaces for increasing the surface area of absorption. The ciliated columnar epithelium is found in the lining of fallopian tubes and parts of the respiratory system, where the beam of the microvilli helps remove particulate matter. Non-ciliated simple columnar epithelium does not have uniform microvilli and helps in secretion and absorption. Pseudostratified columnar epithelium. Pseudo means false and stratified means multiple. Pseudostratified columnar epithelium tissue is a single layer of cells that appears in multiple layers, stratified due to the presence of multi-layered nuclei. The nuclei are located at different levels instead of grouping at the base, thereby giving the look of various layers. However, all the cells rest on the basal lamina. The function of this epithelium is the secretion of movement of mucus. The cells have hair-like projections on the membrane known as cilia that aid in the removal of bacteria or foreign particles by trapping. Example: lining of the airways of upper respiratory tract and movement of egg in the fallopian tube. Stratified columnar epithelium. If present in areas subjected to wear and tear, it protects the underlying tissue as it is present in many layers. It can be further classified as stratified squamous epithelium. In this tissue, the apical layer consists of squamous cells, whereas the basal layer consists of cuboidal and columnar cells that become flattened as they move upwards. The major role is the protection of underlying tissues. Example: lining of the vagina, oral cavity, the throat forms the epidermis, and the top layer of skin. Stratified cuboidal epithelium contains 2-3 layers of cuboidal shaped cells present in the apical layer. These cells aid in protection, secretion, and absorption. Example: the lining of sweat glands, salivary glands, mammary glands, and pancreas. Stratified columnar epithelium consists of 2 or more layers of irregularly shaped cells with apical layer having columnar cells. The function is protection and secretion. Example: lining of the urethra, pharynx, esophagus, and stomach. Transitional epithelium. In the apical layer, there are various types of cells which consist of cube-shaped, on relaxing, and elongated, on stretching. The function is protection and dispensability. Example: inner lining of urinary bladder and lining of ureters and urethra. Figure 7.3. Types of lining and covering epithelium. Simple epithelial tissue is organized in the form of a single layer of cells where all the cells rest on basement membrane, and stratified epithelial tissue is organized as 2 or more layers of cells where only basal layer rests on the basement membrane. Glandular epithelium forms the lining of various glands of the body, such as pituitary glands, adrenal glands, etc. A gland is a specialized structure that has cells epithelial to produce and secrete specific substances chemical into body fluids or ducts. These chemical substances, which is fluid-like, are called secretions and contain proteins. Glandular cells obtain substances from blood and chemically change them by an active process into products called secretion, which are then released from the cells. Based on the secretion of products, glands are classified as endocrine and exocrine. Glands can also be classified based on the number of cells present in them as unicellular and multicellular. Endocrine glands. These glands secrete their products, called as hormones or chemical messengers, directly into the blood or body fluids. All hormonal glands are endocrine glands. The products of endocrine glands, which are released into the interstitial fluid, are diffused into the bloodstream by exocytosis. From bloodstream, the hormones are then delivered to the target cell. Hence, endocrine glands are also known as ductless glands. Endocrine system is crucial for the integration and regulation of bodily responses. Example: hormones produced by intestinal cells cause pancreas to release enzymes that help in digestion. Exocrine glands. The exocrine glands release their products and secretions through ducts that secrete the products into an open space. Examples: sweat glands, oil glands, salivary glands, and tear glands. Based on the number of cells present, exocrine glands can be classified as unicellular and multicellular. Unicellular exocrine glands. These glands release their secretions directly by exocytosis. Unicellular exocrine glands are found within the epithelial lining of intestinal and respiratory tracts. Mucin, a glycoprotein, is produced by these glands, which when dissolved in water, form mucus for protecting and lubricating surfaces. Multicellular exocrine glands. The secretions of multicellular exocrine glands are released on the epithelial surface. Multicellular exocrine glands have more than one cellular unit, an epithelium-derived duct and a secretory unit. The secretory unit is surrounded by connective tissue, which supplies blood vessels and nerves to it. Multicellular exocrine glands are further classified based on the structure of their duct as 1. Simple glands, contains an unbranched duct. 2. Compound glands, contains a branched duct. Compound glands on the basis of secretory units are further classified as tubular, alveolar, and tubuloalveolar. Tubular compound glands have secretory cells that form tubes. In case of alveolar, secretory cells form small sacs. Similarly, tubuloalveolar have both tubular and alveolar secretory cells. Figure 7.4. Modes of secretion. Based on the mode of secretion, multicellular exocrine glands are classified as merocrine glands. The secretory cells of merocrine glands excrete their products by exocytosis without causing any damage to the secretory cells. Salivary glands, pancreatic glands, and eccrine glands are examples of merocrine glands. Apocrine glands. Secretion occurs in such a way that membrane-bound cytoplasm from the apical surface of the cell pinches off into the lumen of the duct. Examples include mammary glands and axillary sweat glands. Polycrine glands. Secretion consists of secretory product along with its own disintegrated secretory cell. The plasma membrane ruptures when the product is released. Example is sebaceous gland present in skin. Figure 7.4 types of multicellular exocrine glands. The multicellular exocrine glands may have simple or branched ducts with secretory units. Connective tissue. Connective tissue is one of the four main types of tissues in the human body. Connective tissue provides structural and metabolic support and is the most abundantly distributed tissue. It serves as a connecting link for binding, strengthening, and supporting all the other body tissues. The main components of connective tissues are ground substance, fibers, and cells. The group substance is clear, viscous, colorless, and fills space between cells. Ground substances constitute the fluid extracellular matrix. Protein fibers constitute the solid extracellular matrix, and together they make up the matrix. The matrix secreted by the connective tissue determines tissue volume and specialized function. Connective tissue is broadly classified into two types: embryonic connective tissue. Meet mature connective tissue, embryonic connective tissue. 1. Mesenchyme. The mesodermal germ layer of the embryo is the source from which all the connective tissue is derived. Mesenchyme is the very first connective tissue that is developed in the embryo, and in the later stages, it is the stem cell line from where all the other connective tissues originate. In adult form, mesenchymal cells are present in irregular, scattered patterns and play a role in the repair and replacement of connective tissue after injury or damage. Example, present under skin in embryo along the developing bones, whereas in adults, along blood vessels. 2. Mucous connective tissue. It is also known as Worden's jelly, consisting mainly of ground substance hyaluronic acid and chondroitin sulfate. It also has low amounts of collagen and reticular fibers. Example, found in umbilical cord. Mature connective tissue. 1. Loose connective tissue. Loose connective tissue helps the surrounding neighboring cells and tissues by allowing the diffusion of water, salts, and various nutrients. It is also present in many tissues, where it prevents shock absorption. 2. Areolar connective tissue. It is the least specialized of the various types of connective tissues. In addition to collagen, elastic, and reticular fibers, it consists of different types of cells: fibroblasts, adipocytes, mast cells, and macrophages. It has an open framework with a major role of filling spaces, muscle fibers, surrounding lymph and blood vessels, and also provides support, strength, and elasticity to the organs in the abdominal cavity. Areolar connective tissue also holds blood vessels and capillary beds. Example, the subcutaneous layer of skin, lamina propria of mucous membrane, around blood vessels, nerves, and organs. 3. Adipose connective tissue comprises of adipocytes, triglycerides, which are the fat storage cells. These fat cells in adults do not divide, however, they expand to store fat. Fat cells are located at the center and consist of a nucleus, cytoplasm, and little extracellular matrix. When the fats are released, the cell shrinks. Adipose tissue is of two types: white fat and brown fat. White fat is the most common, stores fat, absorbs shock, and provides slow heat loss. Brown fat is more vascularized and releases energy when stimulated by the nervous system. Example, found is padding around joints, behind eyeball, in the socket, and surrounding heart and kidney. Figure 7.5. 4. Reticular connective tissue consists of reticular cells and fibers that form a complex and three-dimensional network. Supportive fibers form the stroma of the organs and support functional cells like the parenchyma. It filters the waste product like dead cells in spleen and microbes in lymph nodes. Example, found in reticular organs, spleen, liver, lymph nodes, and bone marrow. Figure 7.6. Figure 7.5. Adipose tissue. A loose connective tissue consists of fat storage fibers and cells. Figure 7.6. Reticular tissue. A loose tissue comprised of a network of reticular cells with little extracellular matrix. 5. Dense connective tissue. Dense connective tissue refers to connective tissue that is packed with large amounts of collagen fibers or elastic fibers. Hence, dense connective tissue can resist stretching to a greater extent. It is further classified as figure 7.7. Figure 7.7. Dense connective tissue. A. Dense regular connective tissue consists of collagen fibers packed into parallel bundles. B. Dense irregular connective tissue consists of collagen fibers intertwined into a mesh-like network. 6. Dense regular connective tissue. Collagen fibers are tightly packed and arranged parallel to each other in bundles with a shiny white appearance. Presence of elastin enables ligaments to get back to their original shape after any stretch. Example, found in ligaments that connect bone to bone, as tendons that attach muscles to bones. 7. Dense irregular connective tissue. Collagen fibers are randomly arranged to form an interwoven network. This network formation provides the tissues with greater strength in every direction. Example, around bones, periosteum, around cartilage, perichondrium, capsules around organs, liver, kidneys, testes, and lymph nodes, dermis of skin, pericardium of heart, and heart valves. 8. Elastic connective tissue consists of elastic fibers that are freely branching. In between the elastic fibers, fibroblasts are present. Example, located in the trachea, bronchial tubes, ligaments between vertebrae, and lung tissue, etc. Cartilage. Cartilage is a supporting connective tissue that is capable of tolerating mechanical stress and absorption of shock. Cartilage is composed of cells and extracellular matrix. The cells of cartilage are chondrocytes, chondroblasts, and chondroclasts. Extracellular matrix consists of ground substance and fibrils. The appearance of cartilage is due to the presence of polysaccharides called chondroitin sulfates. This polysaccharide binds with ground substance proteins to form proteoglycans. Proteoglycans, glycosaminoglycans, and structural glycoproteins are components of ground substance. Perichondrium is a capsule that surrounds the cartilage as a dense sheath. In case of articular cartilage, perichondrium is absent, hence nutrients are supplied to chondrocytes by diffusion through the matrix. The cartilage tissue is of three types. Figure 7.8. Figure 7.8. Types of cartilage. Cartilage is a connective tissue comprised of collagen fibers embedded in a rigid matrix of chondroitin sulfates. A. Hyaline cartilage. B. Elastic cartilage. C. Fibrocartilage. Hyaline cartilage has a bluish-white, translucent appearance and is the most common type of cartilage present in the body. Composed of chondrocytes housed in lacunae, which are matrix cavities. The extracellular matrix is glassy and rich in acidic glycosaminoglycans. Aids in joint movement by providing smooth surfaces, support, and flexibility. Example, walls of larynx, trachea, bronchi, ventral ends of ribs, and temporary skeleton of embryo. Elastic cartilage has a yellowish-colored appearance. Chondrocytes are placed in lacuna as pairs or in single form. Contains fibrils of collagen tubing, extracellular matrix, and fine elastic fibers arranged between the cells. Example, epiglottis, earlobes, eustachian tube, and cartilage in larynx. Fibrocartilage. Chondrocytes are arranged in long rows and appear similar to fibroblast. Extracellular matrix contains thick dispersed collagen fibers. Example, pubic symphysis, intervertebral discs, knee and jaw joints, etc. Bone. Also known as osseous tissue. This is the strongest and hardest connective tissue. It has resistance to shattering due to the presence of flexible collagen fibers. Bone tissue is of two types: compact bone tissue and spongy bone tissue. Compact bone tissue consists of osteons containing lamellae, lacunae, osteocytes, canaliculi, and Haversian canals. Spongy bone tissue is composed of trabeculae, which are thin columns. Space between the trabeculae is filled with red bone marrow. Osteocytes are bone cells arranged within a matrix around the central canals. Both spongy and compact bone provide strength, support, protection, and storage and houses blood-forming tissue. Example, spongy and compact bone tissue forms various parts of bones. Fluid connective tissue. Lymph and blood are the major components of fluid connective tissue. The blood component of fluid tissue has four elements: red blood cells, erythrocytes, white blood cells, leukocytes, and platelets, thrombocytes. RBCs help in transport of oxygen and carbon dioxide. WBCs carry phagocytosis and protect the body against allergic reactions. WBCs play an important role in an immune response. Platelets are important for coagulation of blood. Example, found in blood vessels, arteries, arterioles, capillaries, veins, and venules, and within the chambers of heart. Lymph is the extracellular fluid and is collected from interstitial space. Lymph contains a liquid matrix and white blood cells. Lymphatic capillaries are very permeable and allow larger molecules and excess fluid to enter into them, and then deliver these molecules into the blood, which otherwise could not enter the bloodstream. Thus, lymphatic capillaries help in the transport of absorbed fats away from the intestine and deliver them to the blood. 1. Nervous tissue. Nervous tissue is a highly specialized tissue that coordinates the activities of the body through the secretion of neurotransmitters and conduction of electrical impulses. The two main types of cells of nervous tissue are neurons and neuroglia. Neurons are the functional cells and supporting the neurons are neuroglia, associated with blood vessels and connective tissue. Anatomically, the nervous system is divided into the central nervous system and peripheral nervous system. Central nervous system consists of brain and spinal cord, whereas the peripheral nervous system comprises of nerves and cell bodies of neurons forming ganglia. Functionally, the nervous system is divided into somatic nervous system and autonomic nervous system, which is further divided into sympathetic and parasympathetic. Nervous tissue has the ability to convert a signal to nerve impulse called its action potentials. 2. Muscular tissue. Muscle tissue is made up of contractile cells. It is divided into three main types: skeletal muscle, smooth muscle, and cardiac muscle. Myocytes are the cells of muscular tissue. The difference between the three types of muscular tissue is in the microscopic arrangement and concentration of contractile proteins, actin and myosin. The fibers of muscular tissue have excitability property due to which they can send an electrical wave along the length of their membrane, which is called as an action potential. Figure 7.9. Figure 7.9 types of muscle tissue. A. Skeletal muscle cells possess striation and nuclei located on the periphery. B. Cardiac muscle are also unenucleated and appear striated. C. Smooth muscle cells are unenucleated and have no prominent striations of skeletal muscles. These are striated or voluntary muscles. Microscopic view shows fibers arranged parallel to each other with alternate dark and light bands. Multinuclei are present at the periphery beneath the sarcolemma. Example: attachment between bones and tendons. B. Cardiac muscles. These are striated and involuntary in nature. Fibers are cylindrical, branched and unenucleated, and appear as Y-shaped. These fibers contain intercalated discs with a single nucleus located centrally. Cardiac muscles are present exclusively in the heart and aids in the contraction of the heart synchronously to permit the pumping of blood to different parts of the body. C. Smooth muscles. These are elongated, spindle-shaped, non-striated fibers with a single, centrally located nucleus. They are involuntary in nature and are found in the walls of arteries where they regulate blood pressure to push blood through the circulatory system. They are also present in the smooth muscles of skin, visceral organs, etc. A brief comparison of structure and properties of skeletal, cardiac, and smooth muscle fibers is given in Table 7.1. The different body systems are as follows: nervous system, respiratory system, excretory system, muscular system, endocrine system, lymphatic immune system, integumentary system, digestive system, skeletal system, circulatory system, and reproductive system. The circulatory system. The circulatory system helps to supply oxygenated blood to various parts of the body for the purpose of cellular respiration. This is a body-wide network that interacts with systems of the body. The circulatory system is mainly responsible for the removal of carbon dioxide, maintenance of pH, transfer of proteins, hormones, cells, and other elements of the lymphatic system. Major organs of the circulatory system are the heart, veins, arteries, capillaries, and red blood cells. Heart is the major muscle that pumps deoxygenated blood to the lungs for the purpose of oxygenation. Veins, excluding pulmonary veins, carry deoxygenated blood from different organs to the heart, whereas arteries, excluding tissue histology function location skeletal muscle. Long, tube-shaped, striated, multinucleated cells, peripherally located nuclei voluntary movement, produce heat, protect organs. Skeletal muscles, tongue, eyes, anus, esophagus. Cardiac muscle long, branched cells, single-aligned nucleus, centrally located nucleus. Involuntary pumping of blood. Heart. Smooth muscle, visceral muscle, short, spindle-shaped, unstriated, uninucleated involuntary movement, compressed ducts. Organs, tubes. Aids in peristalsis, involuntary control of respiration, regulates blood flow in arteries by contraction intervals of hollow organs, alimentary canal. Table 7.1 brief comparison of structure and properties of muscle types. The human body systems, structure and functions. Different organs work together to perform a common function, thus constituting an organ system. Human body is comprised of 11 major organ systems. Different organ systems work together and are related to other organ systems to perform different functions that are needed by the body. Pulmonary arteries supply oxygenated blood to all the cells. The respiratory system. Respiratory system comprises of nose, lungs, trachea, bronchi, bronchioles, and alveoli. The main function of this system is to provide oxygenated blood to all parts of the body and remove waste products, such as carbon dioxide. The respiratory tract starts with the nasal cavity and terminates in the smaller alveoli. In addition to breathing, the respiratory system aids in the regulation of blood pH and body temperature. The nervous system. The nervous system is comprised of a highly specialized network of cells that controls all the other systems via electrical and chemical signals. Major organs of the nervous system are brain, spinal cord, and nerves. Nervous system is of two types: central nervous system and peripheral nervous system. The central nervous system comprises of the brain and the spinal cord is the command center for the body. The peripheral nervous system comprises of nerves that connect every part of the body to the central nervous system. Brain is the control center where all the decisions are made, and nerves conduct impulses to specific cells. The overall function of nervous system are to gather and interpret information, respond to information, and maintain homeostasis. The excretory system. The excretory system aids in removal of waste products, such as urea, CO, and water from cellular metabolism. It also functions to filter blood to remove waste products in the form of urea. Major organs include kidneys, ureters, bladder, and urethra. By separating the toxic products, mineral salts, and other waste products, the excretory system helps in maintenance of pH of blood and a stable internal environment homeostasis for the cellular metabolism. The digestive system. With the help of digestive system, complex food in the body is broken into simple, useful nutrients that can be easily absorbed into the bloodstream and used by the body. The digestive system is comprised of various organs connected in a sequence: mouth, esophagus, stomach, small intestine, large intestine, rectum, and anus. In addition, pancreas, liver, and gallbladder are also part of the digestive system. Pancreas secretes digestive enzymes for the breakdown of food and produces insulin for the maintenance of blood sugar levels in the body. Liver produces bile that helps in breakdown of fats. Proteins are broken down into amino acids, carbohydrates into sugars, and fats into fatty acids and glycerol. The endocrine system. The endocrine system controls and regulates various activities of the body like growth, development, reproduction, and metabolism through the production and secretion of hormones. Hormones are chemical messengers which are produced by various glands and released into the bloodstream. After release into the bloodstream, hormones are transported into their target cells. Major organs include hypothalamus, pituitary, thyroid, thymus, adrenal gland, pancreas, ovaries, and testes. The reproductive system. The main function of the reproductive system is to help humans reproduce for the purpose of perpetuation of their species. The female reproductive system comprises of all the necessary organs required to conceive and give birth to a child. The organs include vagina, cervix, uterus, and the ovaries, which produce eggs for fertilization. The male reproductive system consists of organs used for the purpose of procreation. They are penis, testis, epididymis, and vas deferens. The sensory system. Through the organs of the sensory system, one can perceive different types of sensation. Sensation can be classified into two types: general sensations and special sensations. General sensations include pain, touch, temperature, and pressure. Special sensations are hearing, vision, taste, and smell, which carry the sensations to the brain via cranial nerves. Sensory organs include eyes, ears, nose, and tongue. Vital organs. Organs that are necessary for survival are called vital organs and include heart, brain, kidney, liver, and lungs. If any one of the five vital organs stops functioning, it results in the death of a human being. Heart is necessary to keep blood flowing in the body and for the purpose of transporting oxygen, nutrients, hormones, and removal of waste products. Brain is the control center of the body that receives and sends impulses to different regions of the body. It is the place for thought processing, decision making, memory storage, analysis, etc. The two kidneys located on either side of the body, back of the abdomen, are meant to filter blood, form urine, and remove waste products. Liver is an important organ of the metabolic system. It performs a variety of functions like detoxification, blood purification, secretion of bile, removal of bilirubin, production of cholesterol, and some clotting factors. Heart is necessary to keep blood flowing in the body and for the purpose of transporting. It is a place for thought processing, decision making, memory storage, analysis, etc. The two kidneys located on either side of the body, back of the abdomen, are meant to filter blood, form urine, and remove waste products. Liver is an important organ of the metabolic system. It performs a variety of functions like detoxification, blood purification, secretion of bile, removal of bilirubin, production of cholesterol, and some clotting factors. Lungs are located in the upper chest region and work with the heart for purification of blood. The main function is oxygenation of blood and removal of carbon dioxide. The circulatory system. Cardiovascular system consists of the heart and the circulatory system. The heart, a vital organ, acts as a pump to supply blood to all parts of the body, i.e., cells, tissue, and organs. As the blood is circulated in the body, it carries oxygen and nutrients, and at the same time, it removes carbon dioxide and waste products. Veins, arteries, and capillaries carry blood in them from heart to various parts by forming a complex network. Arteries carry purified blood from heart, whereas veins bring pure deoxygenated blood to the heart for purification. Overview of blood. Roughly 8% of an adult's body weight is the amount of blood present in the body. The blood is a slightly alkaline, viscous fluid with a pH of 7.35 - 7.45. The volume of blood in males and females differs slightly. Males have approximately 5 to 6 liters, and females have 4 to 5 liters. Functions of blood. The main function of blood is to help in transportation of oxygen and carbon dioxide in the body. Hormones which are produced in the respective glands need to be transported to the target organ. Hormones are chemical signals released into the bloodstream. The blood then delivers the hormones to the target site. In the digestive system, food is broken down into simpler absorbable substances and transport of these nutrients to various storage sites is facilitated by blood. Blood also works with liver and kidneys to remove impurities and eliminate waste products. Blood is 3 to 5 times viscous than water, meaning it is thicker than water and has resistance to flow. Viscous nature of blood helps in maintaining normal blood pressure. Blood contains proteins known as clotting factors that form clot when there is an injury or damage, thereby preventing blood loss. Specialized cells of blood protect the body against various microorganisms, viruses, infections, diseases, etc. One of the important physiological functions of blood is maintenance of constant temperature, pH, and water balance. Blood volume. The total amount of blood being circulated through the 4 chambers of the heart along with arteries, veins, venules, and capillaries is known as blood volume. Blood volume depends on weight, size, and health condition of an individual. An average human adult contains 5 liters of blood in the body at any time. However, in case of women, the blood volume is slightly lower due to menstrual cycle. Blood volume varies between different organs and needs to be regulated for constant perfusion of blood through body tissues. Factors affecting blood volume include amount of sodium content and hydration status of the body. Blood volume comprises the components of blood, plasma, and various blood cells. The levels of blood volume can increase or decrease if there is any dysfunction in the body, which leads to edema or hypovolemic shock, respectively. Constituents of blood. Blood is a fluid connective tissue that is composed of 60% plasma and 40% blood cells. Plasma, a clear extracellular fluid, contains dissolved substances and plasma proteins. Blood cells, also known as foreign elements, are of 3 types: red blood cells, white blood cells, and platelets. Blood plasma. Blood plasma is a clear straw-colored fluid portion of the blood without the foreign elements. It consists of plasmin proteins, hormones, enzymes, waste products, and gases. Blood plasma is composed of 91.5% water and 8.5% of solute components, 7% of which consists of proteins. The solid ability of plasma enables it to transport nutrients, hormones, carbon dioxide, and waste products. Some of the plasma proteins are albumin. Liver synthesizes albumin, which is the most abundant plasma protein, 60%. Albumins have high colloidal osmotic pressure, which is important for maintaining normal blood pressure and blood volume. Albumins aids in transport of drugs, hormones, and fatty acids. Globulins. Globulins are the second most abundant plasma proteins, 36%. There are 3 types of globulins: alpha, beta, and gamma. Alpha and beta globulins are produced in the liver and help in the transport of fats and fat-soluble vitamins. Gamma globulins are produced by lymphocytes. These are immunoglobulins antibodies. Gamma globulins provide immunity and fight off foreign substances that enter the body. Fibrinogen. Fibrinogen is synthesized in the liver and is important for the clotting of blood. Fibrinogen is a precursor form of fibrin which is important for the formation of the framework of the clot. In addition to the plasma proteins, plasma contains salts less than 1%, aids in metabolism, maintain osmotic balance and blood pressure. Gases, oxygen, and carbon dioxide involved in metabolism and cellular respiration. Nutrients, lipids, carbohydrates, and amino acids serve as food for the cells. Hormones and vitamins, nitrogenous waste, urea, and uric acid. Formed elements. Formed elements are also known as blood cells and are classified as red blood cells, RBCs, white blood cells, WBCs, and platelet. These are enclosed within the plasma membrane and have a definite structure and shape. Platelets are not referred to as cells because they are fragments of bone marrow cells unlike RBC and WBC. Figure 7.10. Packed cell volume, also known as hematocrit, is defined as the total percentage of red blood cells in the blood. For example, hematocrit of 25% means that there are 25 million RBCs in 100 milliliters of blood. Normal range of hematocrit for an adult male is 42% to 54% and for an adult female is 38% to 46%. Normal range of hematocrit varies depending on the age. In case of high altitudes and smoking, hematocrit levels are elevated above normal, and the condition is known as polycythemia. In polycythemia, the viscosity of blood is increased due to abnormal increase in the number of red blood cells. In the embryonic stage, prior to birth, immature precursor cells are converted to mature blood cells by a process known as hematopoiesis or hemopoiesis. In the embryonic stage, hematopoiesis occurs in the yolk sac 3 to 7 months prior to birth. It occurs in the spleen in 4 to 5 months before birth, in the bone marrow cavity. Red bone marrow remains the primary site for hematopoiesis from the 7 months of gestation after birth and continues for the rest of life. In adulthood, blood cell production decreases, whereas in infants, blood cell production is very active. The red bone marrow in infants is replaced by yellow bone marrow on reaching adulthood. Yellow bone marrow consists mainly of fat cells. Red blood cells. Red blood cells, RBC, are also known as erythrocytes. Morphologically, red blood cells have a disc shape with thickened rim and depressed center. Blood type of an individual is decided based on the presence of glycoproteins and glycolipids on the plasma membrane of a mature erythrocyte. Erythrocytes can pass through narrow blood vessels as well as broader vessels due to the presence of special proteins called spectrin and actin on the inner side of the plasma membrane, which make them flexible and durable. These proteins give erythrocytes the ability to stretch, bend, fold in the narrow vessels and regain their normal shape back as they pass through broader vessels. Hemoglobin. The oxygen carrier is present in the cytosol of RCBs. It imparts red color to the blood cells. Hemoglobin constitutes about 33% of the total cell weight with approximately 280 million molecules of hemoglobin present. The red blood cell count in blood is directly proportional to the amount of oxygen being delivered to the tissues in the body. Red blood cell count in a healthy male is 4.7 to 6.1 million cells per microliter cells MCL, and in a healthy female is 4.2 to 5.4 million cells MCL. RBCs lose all their cell organelles, including mitochondria and nucleus during the process of maturation, hence they are incapable of aerobic respiration. As a result, they do not consume the oxygen that is being transported. Due to the absence of a nucleus, they are unable to repair themselves. However, an advantage of absence of cellular components is the greater surface area to volume ratio that facilitates easy diffusion of O2 and CO2 to and from hemoglobin. It also gives the cell a biconcave shape. While passing through capillaries, RBCs undergo wear and tear of the plasma membrane due to which they have a lifespan of 120 days. After a span of 120 days, RBCs get ruptured or hemolyzed. Phagocytic macrophages digest the plasma membrane of the ruptured RBCs to clear them, thereby releasing the hemoglobin into the surrounding medium. The released hemoglobin is either recycled or removed. Erythrocytes perform 2 main functions: carry oxygen and carbon dioxide. RBCs carry oxygen from lungs and deliver it to other parts of the body, and carry carbon dioxide to the lungs for purification. White blood cells. White blood cells, WBC, also called leukocytes, have size of approximately 10 to 14 micrometers in diameter. The main difference between RBC and WBC is the presence of nuclei and absence of hemoglobin in WBCs. Synthesis of white blood cells occur in the bone marrow and maturation in the lymphatic system. Due to the absence of hemoglobin pigment, they appear translucent. Based on the presence of granules in the cytoplasm, WBCs can be classified as granular leukocytes, which include neutrophils, eosinophils, and basophils, and granular leukocytes including monocytes, B and T lymphocytes. Granular leukocytes. Neutrophils, eosinophils, and basophils are the white blood cells containing granules in their cytoplasm. Basophils contain a large basophilic granule and in the granule are present an enzyme, histamine. Histamine is released during allergic reactions to show symptoms like sneezing or runny nose. When stained with basic dye, basophils give a blue-purple color. Eosinophils are enzyme-containing granulocytes that can be visualized as red-orange on staining with acidic dyes. The granules release enzyme to fight foreign substance that has entered the body. Eosinophils kill the invading bacteria, virus, or parasite, and have the ability to promote inflammation. Neutrophils are small and evenly distributed with a pale yellow appearance. The nucleus has 2 5 lobes with an increase in aging, and the older neutrophils have differently shaped nuclear lobes and are called as polymorphic nuclear leukocytes (PMNs). Neutrophils are the first responders to protect the body against infection and injury. A granular leukocytes. Unlike the name given, a granular leukocytes possess granules, but due to poor staining characteristic, they cannot be visualized under light microscope. Monocytes are derived from myeloid stem cells and are the biggest among the white blood cells with kidney-shaped nuclei. They provide protection to the body against pathogens by performing phagocytosis and are hence called macrophages. Astrophilic granules, which are lysosomes, give monocytes the characteristic color and appearance. T lymphocytes and B lymphocytes are the 2 types of lymphocytes. After neutrophils, lymphocytes are the second most numerous white blood cells and are derived from lymphoid stem cell. Morphologically, these cells have a round nuclei and very little cytoplasm. B lymphocytes destroy pathogens by producing antibodies that combine with antigens. Lymphocytes, on the other hand, kill anything directly that has antigens. Some of the important functions of WBCs are defending against pathogens, removal of toxins, waste materials, and damaged cells. Lifespan of WBCs is usually a few days and their number is very less in comparison to RBCs. The normal range of white blood cells is 5,000 to 10,000 cells per microliter of blood. An increase in WBCs count above 10,000 cells microliter blood indicates a protective response of body against pathogens, other conditions, such as exercise and surgery. This condition is generally known as leukocytosis. An abnormal drop in WBCs below 5,000 cells microliter blood is called as leukopenia. Platelets. Thrombocytes, also known as platelets, are membrane-bound cell fragments of bone marrow cells. Platelets are formed from differentiation of hematopoietin stem cells. Myeloid stem cells form megakaryocyte colony-forming cells under the influence of hormone thrombopoietin, which in turn develop into precursor cells called as megakaryocytes. These cells transform into megakaryocytes, which are the large cells that are further fragmented into 2,000 3,000 fragments. Each individual fragment is known as platelet or thrombocyte, lacks nucleus and is surrounded by a membrane. Each platelet is disc-shaped with a diameter of 2 to 4 micrometer. The normal range of platelets in a healthy individual, under normal conditions, is 150,000 to 400,000 platelets per microliter of blood. The life of circulating platelets is approximately 8 to 10 days. Platelets help in preventing bleeding by formation of blood clots. From the site of injury or wound, the damaged vessel sends signals to the platelets for initiating blood clot formation and repairing the damage. Platelets secrete a variety of proteins, blood clot factors for the process of clotting. Thromboxane enhances aggregation of local platelet. Serotonin aids in vasoconstriction, and thromboplastin for blood coagulation. The sticky surface of the platelets enables them to accumulate at the site of damaged blood vessels to form a clot through the process of hemostasis. Abnormal thrombocyte count causes different conditions. When the platelet count in the blood is less due to defect in bone marrow or when the platelets in the body are destroyed, the condition is known as thrombocytopenia. When bone marrow produces excessive platelets, more than 1 million, the condition is thrombocythemia. Thrombocytosis is a condition in which too many platelets are produced due to an underlying infection. Figure 7.10 representation of foreign elements of blood. The foreign elements of blood are classified majorly as erythrocytes, leukocytes, and thrombocytes platelets. The leukocytes can be granular and agranular. Granular leukocytes include basophils, eosinophils, and neutrophils. The agranular leukocytes include B and T lymphocytes. Cell type appearance number of cells per millimeter cube. Functions. Erythrocytes RBCs. Flattened, biconcave disc, red in color, no nucleus 5.2 million, 4 to 4 minus 6.0 million, transportation of oxygen and carbon dioxide. Leukocytes WBCs. Granulocytes neutrophil basophil. Eosinophil. Agranulocytes monocytes. Lymphocytes round in shape, contain a nucleus. Pale yellow, nucleus have 2 to 5 lobes connected by thin filaments 3,000 7,000 phagocytosis of microorganisms and other substances. Stain is blue purple with basic dyes, large violet nucleus. 20 to 50 release histamine to attract and accumulate at the site of inflammation, widens blood vessel. Seen as red orange with acidic dyes, large violet nucleus 100 to 400 kills pathogens, parasitic worms, engulfs antigen-antibody complex phagocytosis. Gray blue cytoplasm with no granules U-shaped nucleus 100 to 700 give rise to macrophages which kills pathogens and damage cell parts. Large round shaped nucleus that almost fills the cell 1,500 3,000 produce antibodies, damages pathogens and disease cells. Thrombocytes platelets. Fragments of megakaryocytes, granules of cytoplasm are stained in purple. 250,000 500,000. Facilitates blood coagulation, enables vasoconstriction by releasing serotonin. Table 7.2 foreign elements of blood. Blood groups. The two major ways to classify the blood groups are the ABO and RH system. ABO system of blood grouping contains 4 blood types, A, B, AB, and O blood types are classified as A, B, AB, and O on the basis of the presence of antigen on the membrane of blood cells. Individuals with type of blood has the presence of antigen A on the red blood cells. Similarly, a person with type B blood has the B antigen. Type AB refers to the presence of both the antigens A and B. Type O indicates absence of both the antigens. The plasma component of blood contains natural antibodies for those antigens not present on the RBCs. Hence, blood type individual has anti-B antibodies circulating in the plasma. Type B individuals have anti-A antibodies. AB blood types have neither of the antibodies, and type O individuals have both the anti-A and anti-B antibodies. Natural antibodies are important during transfusion. For example, if type A individual needs transfusion and type B blood is supplied, then anti-B antibodies circulating in plasma of the individual will clump with the type B antigen present on the membrane of RBCs of the supplied blood. This would lead to a glutination reaction and hemolysis. Blood type O is a universal donor. The RBCs of this blood type do not have any of the two antigens and hence cannot react with any of the antibodies. Rhesus factor or RH factor, also known as antigen D, is another antigen that may be present on the RBCs. RBCs that contain the RH antigen are RH positive, and RBCs without the RH antigen are RH negative. RH negative individuals do not have antibodies to RH antigen. When an RH negative person receives RH positive blood, antibodies will be formed. This may lead to hemolysis on second transfusion as the first transfusion does not cause any problem. Figure 7.11 ABO blood group system with compatibilities between different blood groups. Hemostasis, coagulation, or clotting. Blood clotting is a complex phenomenon involving clotting factors. Blood clotting mechanism is initiated when there is a break in the vessel, which causes a rough surface. The rough surface acts as a stimulus to start clotting within 15 seconds to 2 minutes. Chemicals released during tissue damage, clotting factors, vitamin K, calcium ions, and plasma proteins are involved in the clotting process. Clot formation occurs in a cascade manner with each step leading to the next step in a sequential fashion. The 3 stages of chemical clotting are: Stage 1. This stage is marked by internal damage to the vessel. Factors involved are platelet factors, tissue thromboplastin, factors B, 7, 8, X, X, 11, and 12, and calcium ions. The end product of stage 1 is prothrombin activator. In stage 2, prothrombin is converted to thrombin in the presence of prothrombin, along with calcium ions. In the last stage, thrombin, product of stage 2, converts fibrinogen to fibrin. Other factors involved in the last stage are calcium ions and factor 13. Fibrin is a thread-like protein that forms a mesh-like network to trap RBCs and platelets. This mesh-like network seals the break in the vessel. Once bleeding stops, it is important for the clot to be dissolved. It is known as clot retraction. For clot retraction or fibrinolysis, ATP and factor 13 are important. As the clot is dissolved, platelets break free and release platelet-derived growth factor that helps in the repair of blood vessels. The blood clotting factors involved in the process of blood coagulation are described in table 7.3. Clotting factor description. I-fibrinogen. 2-prothrombin. 3-tissue thromboplastin or platelet factor 3 or extrinsic thromboplastin. I-B calcium. B-labile factor or accelerator. Globulin or proaccelerator B-accelerator. 7-proconvertin. Prothromboplastin 8 antihemophilic factor A AHF or antihemophilic globulin AHG. It is consumed during coagulation and thus not present in plasma. Plasma thromboplastin component PTC or Christmas factor or antihemophilic factor B. X Stewart power factor or thrombokinase present in both serum and plasma. 11 plasma thromboplastin antecedent or antihemophilic factor C. 12 Hagman factor or surface factor or contact factor antihemophilic factor D. 13 Lachy-Laurin factor or fibrin. Stabilizing factor or fibrinase table 7.3 blood clotting factors. Fig. 7.12 mechanism of blood coagulation. When an injury occurs to blood vessel, platelets are activated and stick to damaged vessels and begin the process of blood formation. They interact with blood proteins to form a mesh fibrin which traps the blood cells, thus forming a clot that floods the damage. Anemia. Reduced concentration of hemoglobin in blood or reduced number of blood cells in the normal range RBC, WBC, and platelets is termed anemia. As blood cells aid in oxygen transport, due to anemia the capacity of blood to carry oxygen is reduced. Some of the visible signs and symptoms of anemia are shortness of breath, dizziness, weakness, fatigue, headache, paleness of skin, etc. For an individual to be called anemic, the hemoglobin level should be less than 13.5 gram, 100 milliliters in men and less than 12.0 gram, 100 milliliters in women. However, the concentration of hemoglobin varies with sex, age, pregnancy status, and residence altitude. Children and pregnant women are most commonly affected. Reduced erythropoietic capability can be caused due to the destruction of red bone marrow by harmful chemicals as benzene, by heavy exposure to X-rays, and invasion of bone marrow by cancer cells or chemotherapy for cancer. The common causes of anemia are: the most common type of anemia is iron deficiency anemia. In this type of anemia, there is excessive loss of iron, poor absorption of iron, and insufficient supply of iron to the body. Iron is the major factor affecting. When there is excessive blood loss in the body, it starts to absorb water from tissues outside blood vessels. This results in dilution of blood as additional water is taken up by the blood vessels. Excess loss of blood can occur due to surgery, childbirth, damaged blood vessel, excessive menstrual bleeding, and clot formation. This type of anemia due to excessive blood loss is termed as hemorrhagic anemia. When the synthesis and production of RBCs is reduced, it leads to anemia. It can also be caused due to reduced synthesis of hemoglobin or damage of blood cells. Conditions that cause reduced RBC production are leukemia and sickle cell anemia. During leukemia, production of RBC from stem cells in the bone marrow is reduced. In sickle anemia, the shape of RBCs is changed due to which the oxygen carrying capacity is reduced and can lead to internal bleeding as the RBCs are sickle shaped. Vitamin B12, vitamin K, and folate are important for production of RBCs. Deficiencies in these vitamins lead to less count of RBCs, and this form of anemia is termed as vitamin deficiency anemia. Example: megaloblastic anemia, in which red bone marrow produces large abnormal RBCs. Poor absorption of vitamin B12 in the stomach leads to pernicious anemia. In this form of anemia, the intrinsic factor required for absorption of vitamin B12 is not produced by the stomach. Hemolytic anemia is an autoimmune disorder in which there is abnormal breakdown of red blood cells as the body starts attacking its own RBCs, considering it to be foreign substance. Aplastic anemia is referred to the condition where very few or no stem cells are present in the bone marrow. Example: thalassemia is a rare genetic condition in which red blood cells cannot grow and mature normally. Immunity. Immunity is the capability of the body to resist the infections of the enormous number of cells, molecules, tissues, and organs which are involved in this process constitute the functional system called immune system. Recognition and response are the two functional related activities of immune system. High level of protection is provided by the two systems of immunity that work together, i.e., innate immunity, which is the primitive immune system, and adaptive immunity, which is highly evolved and more specific. Innate immunity. Innate immunity exists from birth. It provides the first line of defense and is non-specific. It responds to almost any kind of foreign organisms and is not specific to any particular pathogen. It is present before the beginning of infection and does not require any earlier exposure to the pathogen. Innate immunity comprises of physical and anatomical barriers, physiological barrier, phagocytic barrier, and inflammatory barrier. Physical barrier includes skin, mucous membrane, saliva, and tears. Skin is the first line of defense. In addition to skin, mucous membrane also inhibits growth of microorganisms. If any pathogens breach the physical barrier, they are exposed to physiological barriers, like temperature, pH, and many chemical mediators, which help in the protection against these microorganisms. Phagocytes are able to surround the particle and engulf it. Macrophages and neutrophils are the examples of phagocytes. These phagocytes are the fast-acting first line of defense when the infectious organisms are successful in invading the physical barriers. Once the physical and chemical barriers are breached by the pathogen, the inflammation, which is a non-specific defense mechanism against the invading microbes or endogenous signals, comes into play. Infection, injury, or disease results in the tissue damage and causes inflammation. This process is represented by the 5 cardinal signs: redness, heat, swelling, pain, and loss of function. Adaptive immunity. Adaptive immunity is the second line of defense. It takes a longer time to develop. The hallmark of adaptive immunity are antigen specificity, diversity, immunologic memory, self-non-self recognition. Unlike innate immunity, which exists even before the beginning of infection and is non-specific, adaptive immunity is highly specific to the foreign antigens and also remembers on its subsequent encounter with a particular microbe that it has earlier infected the body, thereby leading to more rapid elimination of it. Organs of immunity. The most crucial organ which is involved in immune response is the bone marrow. The bone marrow, apart from its role in immune response, bone marrow is also the essence of metabolism, repair, and restoration. It is the site of production of leukocytes that takes part in both innate and adaptive immunity. Red blood cells and platelets are also produced in bone marrow. All the cells of innate and adaptive immunity are matured within the bone marrow. The lymphocyte maturation and function varies between bone marrow and thymus. Natural killer T lymphocytes are matured in thymus, but they take part in innate immunity, whereas the other T lymphocytes are matured in thymus and are part of adaptive immunity. When a body is exposed to antigens which are different from body's own antigens, antibodies are produced against them, and it develops an immunity called as passive immunity. Passive immunization can also be done by injecting antiserum, which contains antibodies that are formed by another person or animal. It provides immediate protection against an antigen, but does not provide long-term protection. Example: tetanus antitoxin is an example of passive immunization. Components of immunity. The immune system includes leukocytes, other chemicals and proteins of blood, such as antibodies, complement proteins, and interferons. Some of them directly attack and neutralize the pathogens, while others work in association to help the immune system cells. Leukocytes, such as lymphocytes B and T lymphocytes, are important components of immunity. B cells become the cells that produce antibodies. The antibodies attach to the antigens and make it easier for immune system to destroy the antigens. The T lymphocytes directly destroy the antigens and help in immune response. T lymphocytes release chemicals like cytokines, which helps in controlling the entire immune response. During the development, lymphocytes learn to differentiate between the body's proteins and foreign antigens. Once B and T cells are formed, few of them divide and provide memory for the body's immune system, which allows the immune system of the body to recognize and respond faster and efficiently to the antigen to which we are exposed again. Inflammation. Once the physical and chemical barriers are breached by the pathogen, the inflammation, which is a non-specific defense mechanism against the invading microbes or endogenous signals, comes into play. Events are involved in the inflammatory process. The bacteria on breaching the physical and chemical barriers damage the tissue and release inflammatory mediators, including vasoactive and chemotactic factors, which help in the increased blood flow in that site and increased capillary permeability. This permits the exudate-containing proteins having antibacterial properties and the phagocytes to enter the site of inflammation and destroy the bacteria. The damaged tissue releases some chemicals like histamine, bradykinin, and prostaglandins. Anatomy of the heart. The heart is a cone-shaped organ located in the thoracic cavity, between the lungs, in the mediastinum. The tip of the heart is downwards and above the diaphragm. The heart is surrounded by a double layer of pericardium, which is a protective layer. Pericardium is made up of 3 layers: fibrous pericardium, parietal pericardium, and visceral pericardium. Fibrous pericardium is the outermost layer made up of a regular connective tissue and interserous pericardium. The space between the outer parietal and the intervisceral layers is called the pericardial cavity. The pericardial cavity is filled with pericardial fluid to reduce the friction between two membranes. The outer layer of pericardium surrounds the base of the main blood vessels of the heart and is attached to the spinal column and diaphragm by ligaments. The inner layer of pericardium is attached to the heart muscle. Epicardium, myocardium, and endocardium are the 3 layers of the heart wall. Epicardium is the outermost layer, made of connective tissue and mesothelium, and has visceral and serous pericardium and myocardium. The middle layer is comprised of cardiac muscle and endocardium. Inner layer consists of endothelium and connective tissue. Chambers of the heart. The blood flows in the internal cavity of the heart, which is divided into 4 chambers. The 2 upper chambers are called left and right atria, whereas the lower 2 chambers are left and right ventricles. Interatrial septum separates the right and left atria and has a depression called fossa ovalis. The interventricular septum separates the right ventricle and left ventricle, and the wall of left ventricle is half-inch thick for pushing the blood through the aortic wall into the body. The walls of atria are thinner in comparison to ventricles that have thick walls. Septum separates the atrial chambers from the ventricular chambers. Due to uneven distribution of myocardium, the thickness of walls of heart chambers varies, and this reflects the amount of force required by each chamber. Impure, deoxygenated blood from the superior vena cava, the inferior vena cava, and the coronary artery is received by the right atrium. The right ventricles receive the blood from right atrium through tricuspid valve. From the right ventricle through the pulmonary valve, the blood is pumped to the pulmonary artery. Oxygen-rich blood from lungs then enters the left atrium from 4 pulmonary veins, 2 from each lung. The oxygenated blood is received by the left ventricle as it exits the left atrium through bicuspid or mitral valve. Finally, the oxygen-rich blood is pumped to the aorta from the left ventricle through the aortic valve, figure 7.13. Circulation of blood. The flow of blood inside the heart chambers is guarded by a set of valves. The opening and closing of the valves is regulated by contraction of the heart chambers as the heart contracts and relaxes due to change in pressure. The main function of the valves is to prevent backflow of blood within the heart. The 2 valves are atrioventricular valves and semilunar valve. The atrioventricular valves AV lies between atria and ventricles. The atrioventricular valves are tricuspid and bicuspid valves. The tricuspid atrioventricular valve lies in between right atria and right ventricle, and the bicuspid atrioventricular valve, also known as mitral valve, lies between left atria and left ventricle on the left side of the heart. Semilunar SL valve is present at the entrance of the aorta, and the pulmonary valve lies at the entrance of the pulmonary artery. In order to prevent flow of blood from ventricles back to atria, the AV valve closes on contraction of ventricles. Semilunar valve closes on relaxation of ventricles to prevent blood from flowing back to the ventricles. Figure 7.13. The internal view of the heart. The right side of the heart pumps blood to the lungs, whereas the left side of the heart pumps blood throughout the body except the lungs. The atria and ventricles are divided by atrioventricular valve, which allows the blood to flow from atria to ventricles. The semilunar valves allow blood to pass out of the heart to different body parts. The right ventricles pump blood through the semilunar valve to the pulmonary artery that is divided into left and right pulmonary arteries, which goes to each lung. The pulmonary veins bring the oxygenated blood back to the heart. The 2 types of circulation are systemic circulation and pulmonary circulation. For systemic circulation, only the left side of heart pumps the blood to the body, except lungs. During systemic circulation, blood from left ventricles is pumped into the aorta and is then carried by the systemic arteries, arterioles, capillaries, venules, veins back to the right atrium. For pulmonary circulation, blood is pumped by the right side of the heart through pulmonary artery into the lungs. Blood is pumped by the right ventricles into pulmonary tank and then it flows into pulmonary arteries, pulmonary capillaries, and into pulmonary veins, which carry blood back to the left atrium. The conduction system. Like smooth muscles, cardiac muscles have single, centrally located nucleus and are striated in nature. Cardiac muscle fibers have numerous blood capillaries for the supply of oxygen and nutrients to the cardiac cells in order to meet their high metabolic demands. Morphologically, cardiac muscle fibers are branched with intercalated discs that connect the branches end to end. The intercalated discs have special structures called desmosomes that provide strength to muscle fiber. Additionally, for the conduction of action potential, gap junctions are present, which allow conduction from one muscle fiber to another. The electrical impulses originated from heart muscle myocardium cause the heart to contract. The sinoatrial SOT node, also called the Z, natural pacemaker, of heart is located at the top of the right atrium and is the starting point for the electrical signals to begin. The electrical impulse from the sinoatrial node causes contraction of muscle fibers of atria and ventricles as it travels through them. Autorhythmic fibers are specialized cardiac muscle fibers that have self-excitable property and can generate electrical action potential by themselves. As these fibers continuously generate action potential, they trigger contraction of heart. Autorhythmic fibers, due to their electrical excitation, cause contraction of heart and hence behave as a pacemaker. They form a specialized network of cardiac conduction system with the use of cardiac muscle fibers to provide direction for cycle of excitation to progress through the heart. The elements of cardiac conduction system are sinoatrial SOT node, atrioventricular AV node, bundle of his AV bundle, and bundle branches. Heartbeat. The human heart has self-excitable property and does not require external stimulation for its contraction and relaxation. It is therefore referred as myogenic heart. For the purpose of blood circulation, the heart undergoes rhythmic contraction and relaxation. When the heart beats, it leads to contraction of the 2 atria. Simultaneously, the 2 ventricles also contract, and then all the chambers relax. The pumping action of the atria and the ventricles lasting about 0.8 seconds is a description of a heartbeat. Sinoatrial node in the natural pacemaker of the heart sends electrical signal every 0.8 seconds for the atria to contract when there is accumulation of blood in the left and right atria, located in the upper chambers. Contraction of heart occurs about 70 times per minute. The specialized fibers of the sinoatrial node cause the heart to beat at a regular interval, at a fixed rate, due to its inherent property. The impulse reaches the atrioventricular node located near the septum at the base of right atria. A 0.1 second delay occurs at the atrioventricular node to allow atria to complete contraction and pump the blood into the ventricles before they begin contracting. After pumping the blood into the ventricles, the atria are empty, without blood in the atrioventricular valves closed to prevent backflow of blood. For ventricular contraction, electrical signal from AV node travels to two branches of AV bundles. Electrical signal from AV node reaches Purkinje fibers, which are specialized neuromuscular cells. Ventricular contraction is brought together by the AV bundle, its branches and Purkinje fibers. The natural pacemaker in the sinoatrial node sends electrical impulse at a regular rate to maintain the heartbeat. However, the heart can still beat if in case the pacemaker stops functioning, but the heart rate will be much slower, 40 to 60 beats per minute. 2. Adrenaline. 2 other hormones, epinephrine and norepinephrine, enhance hormonal control and are released into the blood by the adrenal gland when stimulated by the sympathetic division of the autonomic nervous system. These hormones accelerate the heart rate and the volume of blood ejected during the cardiac cycle during fight or flight response. Cardiac cycle. Myocardium, the cardiac muscle tissue, contains self-excitable characteristics and hence is able to generate electrical signals on contraction. As the electrical signals are generated, a wave of contraction travels to the surrounding cardiac muscle to make the neighboring cardiac cells contract. The cardiac cycle is a rhythmic series of contraction of cardiac muscles and is divided into systolic and diastolic phases. In the diastolic phase, blood is pumped through tricuspid and bicuspid valves to the left and right ventricles, lower chambers of heart due to contraction of left and right atria. This phase is longer than the other phase of the two-part pumping system. The second part of the two-part pumping system begins when the ventricles are full. The ventricles contract when the electrical signal. The sonode located at the dorsal wall of the right atrium sends the electrical signal, which causes the atria to contract. The impulse then reaches the AV node, which is located at the base of the right atrial nervous septum. The electrical signal for ventricular contraction from AV node travels to two branches of AV bundles. Finally, the electrical impulse reaches specialized neuromuscular cells called Purkinje fibers. The AV bundle, its branches, and Purkinje fibers altogether bring ventricular contraction. The sonode keeps releasing the electrical impulse at a regular rate which maintains the heartbeat. Nervous and hormonal regulation. Neural, intrinsic, and hormonal factors control the rhythmic beating and impulse conduction within the heart. The control center for heart and lungs is the medulla oblongata, which is responsible for the regulation of heartbeat. In addition to medulla oblongata, the heart rhythm and the cardiac cycle is regulated by an autonomic nervous system. Adrenaline hormone secreted by the sympathetic nervous system increases the heart rate, whereas the parasympathetic nervous system secretes acetylcholine hormone, which reduces the heart rate contrary. Figure 7.14 electrical impulses through the heart. In case the sinoatrial node fails to function, atrioventricular node generates impulses to keep the heart functioning. Else, an artificial pacemaker is implanted to generate the electrical impulses every 0.8 seconds. Figure 7.14. From the sonode travels along the ventricular walls resulting in the systolic phase. To prevent blood from ventricle to flow back to atria, the tricuspid and bicuspid valves close. In a similar way, to push the blood to lungs, the pulmonary and aortic valves open. The deoxygenated blood is pumped to lungs from right ventricle, where blood picks oxygen, and oxygen-rich blood is pumped from left ventricle to heart and other parts of the body except lungs. Once the blood is pumped into aorta and pulmonary artery, the aortic and pulmonary valves closes. The lower pressure in ventricles causes the tricuspid and mitral valve to open and another cardiac cycle begins. This series of contractions is repeated over and over again. The heart normally beats around 70 times in a minute. Atrial systole and diastole. Depolarization of atrial muscle happens when the atrial muscle contracts from superior to inferior portion of atria towards atrioventricular septum. The pressure within the atria rises. As the pressure is increased, the bicuspid and tricuspid valves of the atrioventricular valve open and the blood is pumped into the ventricles. In an ECG, the P wave stands for atrial systole. As the atrial systole begins, the ventricles are approximately 70-80% filled with blood, whereas the remaining 20-30% of the ventricular volume is filled with blood by atrial kick, also known as atrial systole. This phase lasts about 100 milliseconds and ends prior to ventricular systole, as the atrial muscle returns to its diastolic phase. Ventricular systole. In an ECG, QRS complex represents the ventricular systole. This phase follows ventricular depolarization. In this phase, the atrioventricular valves close to prevent the backflow of blood into atria as the pressure rises in the ventricles. During the initial ventricular systole, the pressure inside the ventricles is not sufficient to eject the blood from the heart to the vessels. However, the pressure quickly rises above the atria that are now in a relaxed state, which shuts the atrioventricular valves. Since the blood is not being ejected from the ventricles, in early ventricular systole, the volume of the blood within the ventricles remains constant. This phase of ventricular systole is termed as isovolumic contraction. In later stage, the increased pressure in ventricles forces the semilunar valves in aorta and pulmonary artery to open to make the entry of blood into these vessels. The closing of atrioventricular valve during ventricular systole produces the first heart sound love. This marks the beginning of ventricular contraction, ventricular systole. Ventricular diastole. T wave in an ECG stands for ventricular diastole, which refers to repolarization of ventricles. In this phase, the ventricular muscles start relaxing and the pressure within the ventricles drops below the pressure in bot, the pulmonary trunk, and aorta. The semilunar valves remain closed to prevent backflow of blood into the heart. The atrioventricular valves also remain closed at the early stage of ventricular diastole. Thus, there is no change in the volume of blood in the ventricles and is generally termed as isovolumic ventricular relaxation phase. During the late ventricular diastole, as the ventricular muscles relax, the pressure within the ventricles drops even further. Atrioventricular valves open and push the blood into ventricles when the pressure in the ventricles falls below the pressure in atria. This results in closing of semilunar valves. As the semilunar valve closes, it produces a second sound of heart up. Cardiac cycle duration. The time duration of atrial cycle is divided into atrial systole and atrial diastole. Atrial cycle. Atrial systole equals 0.1 seconds, atrial diastole equals 0.7 seconds. The time duration of ventricular cycle is divided into ventricular systole and ventricular diastole ventricular cycle. Ventricular systole equals 0.3 seconds, ventricular diastole equals 0.5 seconds. The atria and ventricular systole occur independently of each other. Their time durations are also calculated separately. But diastolic phase of two cycles occur at the same time for 0.4 seconds. Therefore, the time duration is taken once and referred as joint diastole. The time duration of one cardiac cycle is calculated as follows. Atrial systole equals 0.1 seconds, ventricular systole equals 0.3 seconds, joint diastole equals 0.4 seconds, total equals 0.8 seconds. Blood pressure. The blood pressure is defined as the pressure of circulating blood exerted on against the walls of the arteries when heart beats. Blood pressure readings have two measurements, of which the top number is systolic blood pressure, which is referred to as the highest pressure during the time heart beats and pumps blood around the body. The bottom one is diastolic blood pressure, which is the lowest blood pressure when heart relaxes in between the beats. The blood pressure is expressed as the ratio of systolic and diastolic pressure. The blood pressure is measured by using sphygmomanometer in the brachial artery of the arm. The normal resting blood pressure for a young adult is considered to be 120 eightieths of a milliliter Hg. The higher value represents the systolic pressure, and the lower value represents the diastolic pressure. In case of high blood pressure, the systolic pressure is 120 to 129 Hg, and the diastolic is less than 80 milliliters Hg. Both systolic and diastolic pressure increases with age. The systolic pressures increase more than the diastolic pressure due to stiffness of arteries following arteriosclerosis with increasing age. When the systolic pressure ranges from 130 to 139 Hg, and the diastolic ranges 80 to 89 Hg, the condition is called as hypertension stage 1. Hypertension stage 2 is marked by consistent blood pressure range of 140 90ths of a milliliter Hg or higher. Cardiac output. For the body to function properly, the heart needs to pump blood at a sufficient rate to all the vital organs of the body. The blood supplies oxygen and nutrients to the vital body organs for their normal functioning. The cardiac output milliliter per minute can be described as the amount of blood heart pumps every minute. In a resting state, a healthy individual's heart with a normal cardiac output generally pumps about 5 to 6 liters of blood every minute. During exercise, the muscles need more oxygen, thus the cardiac output increases. The heart beats faster during exercise so that more blood is pumped from heart and supplied to the body. The stroke volume ml beat is the amount of blood pumped at each heartbeat, and the heart rate beats per minute is number of times the heart beats per minute. Body organ amount of blood flow. Milliliter per minute. Muscles 1000, skin 300, brain 700, kidneys 1100, liver 1350, others 350. Table 7.4 cardiac output distribution. Electrocardiogram ECG. Electrical changes generated by the cardiac muscle tissue and myocardium during the cardiac cycle can be measured by placing electrodes on the surface of the body. Such recording is called as electrocardiogram, and the instrument used for the measurement of electrical activity is electrocardiography. Body fluid contains ions. These ions conduct electrical signals that can be recorded on the surface of skin. Electrodes in the form of plastic patches are placed on the chest, arms, and the legs. These patches are connected through lead wires by an instrument ECG machine that records the electrical changes in the myocardium. Figure 7.15. ECG contains three different deflections or waves. P wave. Electrical impulses that travel from the sinoatrial node through the right and left atria that result in the pumping of blood into ventricles are recorded in the P wave, representing depolarization of the atria. The P wave lasts usually 80 to 100 ms. PR interval. Electrical impulses travel from atria to the ventricles through the atrioventricular node. As the impulse passes to the ventricles, there is a delay in time, and the signal is recorded at the end of P wave and the beginning of Q wave is a straight line. The normal range of PR interval in adults is between 120 to 200 ms. QRS complex. Electrical impulses reach the ventricular myocardium through a bundle of His, depolarize, thus pumping blood to the lungs. The impulse is recorded as QRS complex and represents depolarization of the ventricles. The duration of QRS wave in normal, healthy adults is 60 to 100 ms. However, physiological factors can affect T wave. It represents repolarization of the ventricles. As the blood is pumped into the lungs, the ventricles start to repolarize, relax, and this is recorded as T wave. Atria gets filled with blood again to start the next cycle. ECG is helpful for the diagnosis of valve-related disorders, atherosclerosis, hypertension, and rheumatic fever to name some. Figure 7.15 representation of a typical electrocardiogram. ECG. ECG is a measure of electrical activity of atria and ventricles. Blood vessels. Arteries, veins, and capillaries are the blood vessels of the circulatory system. Arteries and arterioles transport blood to the capillaries from the heart. Veins and venules transport blood from capillaries to the heart. Capillaries are involved in exchange of gaseous materials with the cells and tissues. Arteries. Arteries are efferent vessels that carry oxygenated blood, except pulmonary arteries, away from heart to capillaries. Pulmonary arteries carry the oxygenated blood for purification from heart to lungs. Arteries are made up of tunica intima, tunica media, and tunica adventitia. The innermost layer, tunica intima, has endothelial lining of connective tissue and elastic tissue, which permits the flow of blood easily. Arteries are efferent vessels that carry oxygenated blood, except pulmonary arteries, away from heart to capillaries. Pulmonary arteries carry the oxygenated blood for purification from heart to lungs. Arteries are made up of tunica intima, tunica media, and tunica adventitia. The innermost layer, tunica intima, has endothelial lining of connective tissue and elastic tissue, which permits the flow of blood easily. Tunica media, the middle layer, has smooth muscles and thick connective tissue, helping in pumping of blood. The outermost layers are tunica external or adventitia with collagen fibers and smooth muscles and small blood vessels. Arteries are divided into small branches called as arterioles that are supplied with autonomic nerve fibers. Due to the elastic nature of arteries, they can withstand pressure during the contraction of ventricles. Capillaries. Capillaries are thin-walled, composed of a layer of endothelial cells. They connect the arteries and veins and provide large surface area for gaseous exchange and waste exchange. Capillaries get oxygenated blood from arteries, and exchange of carbon dioxide and oxygen occurs at capillaries. Capillaries then deliver the oxygenated blood to veins which further deliver it to lungs and heart. The different types of capillaries are continuous, fenestrated, and sinusoids. Continuous capillaries are found in muscle cells. Fenestrated capillaries have pores between adjacent endothelial cells, found in kidneys and endocrine glands. Sinusoids capillaries are not continuous, found in liver and spleen, and have membrane-covered gaps between endothelial cells. Capillaries are specialized for exchange of materials with interstitial fluid. Veins. Small venules unite to form veins. Deoxygenated blood from venules is drained into the right atrium by veins via the vena cava. Oxygenated blood from the lungs is taken by the pulmonary veins to the left atrium, whereas blood from the capillaries of the gut is taken by the hepatic portal vein to the capillaries of the liver. The makeup of veins is the same as the tissue layers of arteries. However, the middle layer is thinner, lesser elastic tissue and muscle, therefore makes the veins compressible and distensible. Blood pressure in veins is lower than that of arteries due to thinner walls and larger lumen. Some veins and legs possess valve in them to prevent the backward flow of blood, but when these valves leak, the backward flow of blood causes the veins to stretch and get elongated. Routes of circulation. The different circulatory paths of the cardiovascular system are systemic and pulmonary circuits of circulation. Systemic circulation. In systemic circulation, deoxygenated blood is brought back to the heart while the oxygenated blood from heart is carried to the rest of the body for transport of oxygen and nutrients. Oxygenated blood from pulmonary veins enters the left atrium, then to the left ventricle through the bicuspid valve. From left ventricle, blood is pumped into aorta through aortic valve. Aorta branches into arteries, which further branch into arterioles and capillaries. Deoxygenated blood from capillaries reaches vena cava through veins and venules, and finally drains into the right atrium. From right atrium, pulmonary circulation starts so that blood can be oxygenated before reaching systemic circulation again. The systemic circulation is further subdivided into coronary, cardiac, and hepatic portal circulation. The systemic circulation mainly functions to transport oxygen to vital organs and other cells of the body, along with removal of waste products and carbon dioxide. Coronary circulation. Coronary circulation refers to the flow of blood in the coronary arteries of the myocardium. Coronary arteries deliver oxygenated blood to the myocardium, and the cardiac veins remove the oxygenated blood from the myocardium. This path of circulation protects the heart tissue from damage. The rate of blood flow by coronary arteries to the heart muscles is 200 milliliters per minute. The maximum amount of oxygen available, 70–75%, is extracted by the heart, much more than any other organ. Hence, obstruction of coronary arteries results in damage or death of heart muscle as oxygen is not available to the heart tissue. This condition is known as myocardial infarction or heart attack. Another condition called angina pectoris occurs when there is a partial blockage of coronary artery, the sign of which is pain in the left arm. Hepatic circulation. Portal refers to the vessel that carries blood from one capillary network to another. Hepatic portal system is the venous system that carries the oxygenated blood from gastrointestinal tract and spleen to the liver. Capillaries for hepatic circulation are located at the villi of small intestine and inside the liver. Hepatic portal vein is formed when the capillaries of the intestine combine with the venules. Pulmonary circulation. Pulmonary circulation is the circulation of blood for the purpose of oxygenation from heart to lungs, back to heart. The oxygenated blood leaves systemic circulation on reaching right atrium through the superior and inferior vena cavae. Blood is transported to the right ventricle through the tricuspid valve, and then to the pulmonary artery through pulmonary valve. From left and right pulmonary arteries, blood travels to left lung and right lung, respectively. In lungs, blood reaches alveoli, the site of gas exchange where carbon dioxide from blood is eliminated and oxygen is added. This oxygenated blood then passes through the pulmonary venules, which reaches left atrium via pulmonary veins. Figure 7.16. A brief overview of systemic and pulmonary circulation is given in table 7.4. Figure 7.16 representation of routes of circulation. Left side of heart pumps blood for pulmonary circulation, and right side of heart pumps blood for systemic circulation. The deoxygenated blood is pumped to lungs through pulmonary arteries and picks up oxygen in lungs and is returned to heart via pulmonary veins. Systemic circulation. Pulmonary circulation. Source left ventricle, right ventricle termination, right auricle arteries typical. Mean pressure. Vessel wall. Oxygen content. 100 milliliters of mercury thick and elastic. High. 15 milliliters of mercury. Thin and non-elastic. Low. Veins. Typical mean pressure. Oxygen content 2 milliliters of mercury low. 5 milliliters of mercury high. Table 7.4 overview of systemic and pulmonary circulation. Lymphatic system. Unlike the cardiovascular system, the lymphatic system is not closed and is a specialized system. Lymphatic system through the capillaries facilitates diffusion, and it consists of lymphatic vessels and organs. The blood that flows through the capillaries loses fluid by the process of diffusion, and the amount of liquid lost due to diffusion is more than the fluid absorbed in the body. In a human being, approximately 3 liters of fluid leaves the cardiovascular system, which is more than half the total supply of body. To balance this loss of this fluid, body uses another circulatory system, open-ended, which collects liquid and returns them back to the cardiovascular circulation. The lymph capillaries collect fluid and carry them to two lymphatic larger vessels, which resemble veins and possess series of one-way valves to facilitate one-way movement of fluid. These two lymphatic vessels drain the fluid to lymphatic veins through one-way valves. The lymphatic vessels are interrupted at intervals by little masses of lymph tissue, called lymph nodes that eliminate the foreign materials as pathogens from the lymph filtering through them. The various functions of the lymphatic system are as: the lymphatic system helps to protect the body against infections and diseases by producing WBCs, thus a crucial part of the body's immune system. The lymphatic system organs and tissues are the major sites of production, differentiation, and proliferation of T and B lymphocytes. The lymphatic capillaries collect the excess fluid and return it to the bloodstream. The lymph fluid carries the waste products and kills bacteria back to the blood where these are eliminated by liver or kidneys. Lymph is the interstitial fluid that flows in between the cells and tissues. It is yellowish in appearance due to the absence of hemoglobin. It transfers materials from blood to body and vice versa. Respiratory system. Introduction to respiration. The energy needed by animals for regular activities is chiefly procured from the oxidative breakdown of foodstuffs, particularly carbohydrates and fats. Dealing with this process, which is called internal or cellular respiration, oxygen O2, which we need for our cells to function, is employed by mitochondria, and carbon dioxide is generated. The oxygen required for this process is eventually derived from the atmosphere by the process of external respiration, which also serves to remove a waste product produced by cells, namely carbon dioxide (CO2). The process that governs external respiration and determines its efficiency is the subject of this chapter. Human respiration. The respiratory system is an interdependent system that consists of the upper respiratory tract and the lower respiratory tract. The upper respiratory tract includes parts outside the chest cavity, nasal passages, nasal cavities, pharynx, larynx, and upper trachea. The lower respiratory tract consists of parts present within the cavity of the chest, lower trachea, lungs, bronchial tubes, and alveoli. In addition, diaphragm and intercostal muscles are also part of the respiratory system. The process of respiration happens in two zones, namely, respiratory zone composed of the bronchioles, alveolar ducts, and alveoli, site of gas exchange. The second zone is conducting zone, includes all respiratory passageways and provides air for exchange of gases. Upper respiratory tract. The inhaled air is warm and humidified because of the vascular and mucoid nature of the nasal cavity. A turbulent gas flow is created in the nasal concha to maximize exposure of the surface area. Epithelial cells are lined with cilia that help infiltration of air in association with the mucus, approximately 1 liter per day. Pharynx, a muscular tube that transports food to esophagus and air to larynx. Secondary function, such as sound production is performed by larynx, homeostasis by lungs, and smell is performed by lungs. Lower respiratory tract. As air is inhaled, it first passes through the larynx and then to trachea, the main conducting tube. Trachea divides into two bronchi, which further divide into branches forming terminal tubes known as bronchial tubes. These tubes terminate at the end forming alveoli, figure 7.17. Nearly 300 million alveoli exist in a pair of human lungs, with each alveolus having a diameter of 75 to 300 micrometers where exchange of gases takes place. After gaseous exchange is done, eliminated out through nose or mouth with the start of exhalation. Exhalation consists of vocal cords present in the larynx for sound production, lungs for homeostasis of body pH levels, and the olfactory bulbs in the nose for the smell. Respiratory physiology employs a large number of extended abbreviations, which are often used to calculate respiratory data. The most common are given below in the table 7.5 together with the conventions for the use. Figure 7.17 diagram of branching airway schematic representation of the division of the airway in the lung from trachea to alveoli. General variables modifying symbol C compliance alveolar gas C concentration ATPD gas at ambient. Temperature and pressure. Dry D diffusion capacity ATPS saturated gas at the surrounding temperature. And pressure ERD expiratory reserve volume B barometric. F respiratory frequency BTPD gas at body temperature and pressure. Dry F fractional concentration. Dry BTPS gas at body temperature and pressure. Saturated F forced expiratory flow D dead space gas FRC forced expiratory volume D expired gas IC functional residual capacity I inspired gas or inspiratory reserve volume L transpulmonary P gas pressure STPD gas at standard. Temperature and pressure. Dry Q volume of blood T tidal gas. Q volume of blood per unit time perfusion. TM transmural. R gas constant of airway R resistance E chest wall R respiratory exchange ratio S esophageal R Q respiratory quotient intrapleural RD residual volume RS trans respiratory system T absolute temperature of gas arterial blood T surface tension B blood TGB thoracic gas volume C capillary blood TLC total lung capacity N number of moles of gas V gas volume P pulmonary B gas volume per unit time. Ventilation. R radius of curvature. BC vital capacity T tissue. V venous blood table 7.5 symbols in respiratory system. Thus, PaO2 equals partial pressure of oxygen in alveolar gas PaO2 equals partial pressure of oxygen in arterial blood. VO2 equals O2 consumption per unit time FeCO2 equals fraction of CO2 in dry. Expired gas VA equals ventilation of alveoli per min sec VA equals ventilation perfusion ratio VAQ. Breathing mechanism. Mechanism of breathing. Ventilation is the movement of air from into the alveoli. Ventilation consists of two aspects, inhalation and exhalation, which is caused due to changes in pressure inside the thorax. Inhalation and expiration are brought about by medulla of the nervous system along with respiratory muscles diaphragm, external and internal intercostal muscles. Diaphragm, located below the lungs, is a dome-shaped muscle. Diaphragm flattens, moves downwards, expanding the volume of chest cavity on contraction. The external intercostal muscles pull the ribs upward and outward, and the internal intercostal muscles pull the ribs downward and inward. Inspiration is when air flows within the lungs. Expiration is when the air is expelled outside, facilitated by the chest muscles, air pressure, and the change in the diaphragm size. Three important pressures in breathing are atmospheric pressure, intrapleural pressure, and intrapulmonic pressure. Atmospheric pressure is the pressure of air in the surrounding. It is 760 millimeters of mercury. Atmospheric pressure and higher altitude are inversely proportional. Space potential not real between the parietal flora and visceral flora is known as pleural space. Pressure within the pleural space is called the intrapleural space and is always less than the atmospheric pressure, about 756 millimeters of mercury, and is called the negative pressure. Negative respiratory pressure minus 4 millimeters Hg indicates that the pressure is lower than atmospheric pressure by 4 millimeters Hg. 760 to 4 equals 756 millimeters Hg. When the pressure is higher than atmospheric pressure, it is termed as positive respiratory pressure. Zero respiratory pressure is equal to atmospheric pressure. Intrapulmonic pressure, intrapulmonary and intraalveolar pressure is the pressure within the alveoli and bronchial tree. This pressure rises and falls during the breathing. Figure 7.18 intrapulmonary and intrapleural pressure. Relationships difference in pressure relative to atmospheric pressure, 760 millimeters Hg, is given in parentheses at the end of a normal expiration. For illustration, the size of the pleural cavity has been greatly exaggerated. Pulmonary ventilation. Pulmonary ventilation or breathing is the process of flow of air into lungs during inhalation or exhalation. This process relies on change in volume in the chest cavity, which affects the pressure change leading to flow of gases to equalize the pressure. Boyle's law addresses the correlation between the pressure and volume of a gas. P1 V1 equals P2 volts 2. Where P1, initial pressure of gas, P2, final pressure of gas, V1, initial volume of gas, V2, final volume of gas. Physical factors influencing pulmonary ventilation. During inspiration, lungs expand or stretch, and during expiration, lungs relax or withdraw. This process of stretching and relaxing consumes energy for enlarging thorax as well as to subdue factors such as airway resistance. The primary resistance to gas flow is friction encountered in the respiratory pathways. The following equation gives the relationship between gas flow F pressure P and resistance R F equals increment P. R gas flow and pressure gradient. Increment P is directly proportional and inversely proportional with resistance. During respiration, the average pressure gradient is 2 millimeters Hg or less, sufficient for moving 500 milliliters of air. However, airway resistance is irrelevant as the airway diameter in the initial part of the conducting zone is massive enough for the low viscosity of air. Furthermore, as the airways get smaller and smaller, it results in vast cross-sectional area. Consequently, as the gas flow stops and diffusion takes over at the terminal bronchioles, resistance is no longer an issue. Alveolar surface tension. Liquid molecules interact greatly with each other compared to gas molecules. This unbalanced attraction creates a surface tension as these water molecules do not interact with gas molecules inhaled, resisting any force which enhances the surface area of liquid. As alveolar walls are coated with liquid film, a surfactant that separates the water molecules from binding to itself and enables it to interact and pass through the airway. This reduces the alveolar surface tension, preventing alveolar collapse. Pressure-volume relationship. A pressure-volume curve is utilized to understand the lung deformation during breathing. It describes the mechanical behavior of the lungs and chest walls, such as the elasticity, ability to expand, and stretch distensibility. This can be obtained with the help of the slope of the pressure-volume curve, figure 7.19, known as lung compliance CL, and has units of milliliter per centimeter H2O equated as LC equals increment V. Increment P where increment V equals change in lung volume, increment P equals change in the pressure. The human lung compliance is 0.15 L per centimeter H2O. When the human lung compliance is high, it means that the lung is easily distended and results in a steep pressure-volume curve. When the lung compliance is low, it means that the lung is stiff, with the pressure-volume curve being flat. Hysteresis is defined as the differences in the pressure-volume curve during inflation and deflation. Figure 7.19, the pressure-volume relation for a single respiratory cycle. The slope of the line can determine the lung compliance. During inspiration, additional pressure is needed to overcome the airflow resistance and other resistive forces. Curve on the right of the compliance. The curve to the left of the compliance line shows the resistive work done during expiration. When reading the compliance slope, the lung volume must also be considered as at low lung volumes. The lung distends easily, but at high lung volumes, there is hardly any change in the lung volume. Lung volumes and capacity. Spirometers are the traditional tools of the respiratory physiologists used to measure the lung compliance. It consists of an inverted bell consisting of a water seal that forms an airtight chamber. The bell moves freely in the vertical direction, and the movements are recorded on a chart spirogram or logged by a computer. Figure 7.20. The subject breathes through the mouth via the flexible tube. Inspiration draws air from the bell, which goes down and decreases the volume of the air trapped within the bell, and increases during expiration. The volume changes are recorded on a calibrated chart or by a computer. The relationship between various lung volumes and typical tracing measured from a spirometer is shown in figure 7.21 and figure 7.22. Respiratory capacities include the tidal volume TD, inspiratory reserve volume RD, expiratory reserve volume ERD, and residual volume. Respiratory capacities can be divided into total lung capacity TLC, vital capacity VC, inspiratory capacity IC, and functional residual capacity. Both the respiratory volumes and respiratory capacities vary between male and female. Dead space and minute ventilation. At rest, a healthy human being inspires approximately 500 milliliters tidal volume of air per breath, and on an average we breathe 15 times per minute. The volume of air we breathe per minute is known as minute ventilation. Thus, minute ventilation equals tidal volume 500 milliliters times breathing rate 15 equals 7,500 milliliters per minute, 7.5 L per minute. Broadly, the respiratory system can be considered to consist of two parts: the conducting airways and the area of gas exchange. In dividing the respiratory system this way, it becomes obvious that not the entire air taken in reaches the alveolar surface. Some of it must occupy the airways that connect. Figure 7.20 recording spirometer. The respiratory surface to the atmosphere. Some volume 150 milliliters of the BT 500 milliliters is left behind, which does not contribute to diffusion known as dead space. This means only 350 milliliters is involved in alveolar ventilation. Thus, by calculation, alveolar ventilation equals 515 ml times 15 equals 5,250 milliliter per minute. Since not all of the air that enters the alveoli takes part in gas exchange, two different types of dead space are distinguished. Figure 7.23. 1. Anatomical dead space. Fowler's method measures the volume of air that is inhaled into the conducting airways but does not contribute to the diffusion in alveoli. This is termed as the anatomic dead space. 2. Physiological dead space. As dead space does not contribute in diffusion, Bohr's understands that dead space cannot contribute in carbon dioxide exhalation. As Bohr's method focuses on areas that are actually involved in gas exchange, the dead space volume includes the conducting airways plus any non-perfused alveoli, and is referred to as a physiologic dead space. Physiological dead space can be estimated by measuring the carbon dioxide content of the expired and alveolar air using the Bohr method. Dead spaces are those areas that do not contribute to gas exchange. Anatomic dead space includes the non-respiratory airways. Physiologic dead space includes the anatomic dead space plus any alveoli that are not perfused and thus cannot participate in gas exchange. Figure 7.21 Summary of respiratory capacities and respiratory volumes. Figure 7.22 Static lung volumes and capacity tracing measured by spirometer. The vertical axis is the volume and the horizontal axis is time. Figure 7.23 Anatomic and physiologic dead space. Comparison. Our tidal volume is calculated as follows. Tidal volume versus T equals dead space BD 500 to 150 plus volume of air entering the alveoli. BT equals BD plus VA. Where VA, volume of alveolar gas during tidal breathing. BT, tidal volume. BD, dead space volume. The tidal volume BT multiplied by frequency F of breathing is known as the minute volume. The part of the minute volume that goes in the alveoli is known as the alveolar ventilation. Therefore, FBT equals FBD plus FVA. 4. BD equals BD plus VA. Where BD, minute volume. BD, dead space. VA, alveolar ventilation. F, frequency. Gas exchange in blood, lungs and tissues with internal and external respiration. O2 enters, and CO2 leaves the blood, respectively with the help of diffusion. The composition and physical properties of gas helps us to understand the above process. Basic properties of gases. 1. Dalton's law of partial pressures. According to this law, the total pressure utilized by the mixture of gases is equal to the sum of the individual pressure exerted by each gas partial pressure in that particular mixture. Moreover, partial pressure is directly proportional to the percentage of those gases in the gas mixture. 2. Henry's law. Henry's law explains the movement of gases. Once a gas comes in contact with a liquid, the gas dissolves and follows the partial pressure of the liquid. If the concentration of the gases is high, it goes inside the liquid at a higher rate. When this partial pressure becomes greater in liquid compared to one in the surrounding gas phase, some of the gases in the liquid will return back to its gaseous phase. Thus, the partial pressure determines the movement and amount of gases in the two stages. Composition of alveolar gas. The alveolar gases are comparatively different than the atmospheric gases. While the atmospheric pressure contains O2 and N2, alveolar gases are composed of CO2, water vapor, and less O2. The difference occurs due to the humidification of air by respiratory and conducting airways, gas exchange taking place in lungs, and the mixing of gases present in alveoli. As the amount of inspired air is nearly 500 milliliters, the final composition of the gases in alveoli is a mixture of new and old gases already present in the respiratory tract. As the breathing intensity increases, it affects the partial pressure of O2 and CO2 in the alveoli. External respiration. During pulmonary gas exchange, external respiration, at the pulmonary circuit, dark red blood is transformed into scarlet color. The systemic arteries return the scarlet colored blood to the heart for it to distribute to the body. This change of color takes place due to O2 uptake, which binds to hemoglobin in red blood cells, RBCs, while unloading of CO2 occurring faster. Following factors affect the flow of these gases. 1. Gas solubilities and partial pressure gradients. Diffusion in the respiratory membrane takes place due to the partial pressure of O2 and CO2. The partial pressure of O2 of the oxygenated blood is only 40 milliliters Hg, in contrast to that of 104 milliliters Hg in the alveoli in the pulmonary circuit. This causes the oxygen to diffuse rapidly from the alveoli into the pulmonary capillary blood. Figure 7.24. Equilibrium PO2 of 104 milliliters Hg on both sides of the respiratory membrane typically occurs within 0.25 seconds, which is 33% of the time an RBC is in a pulmonary capillary. In this way, blood can move through the respiratory vessels 3x rapidly and still be adequately oxygenated. Carbon dioxide diffuses in the opposite direction along a partial pressure gradient at around 5 milliliters Hg until equilibrium happens at 40 milliliters Hg and is then dismissed out slowly. In common, CO2 is 20x more dissolvable in plasma and alveolar fluid than that of O2. So, regardless of the O2 pressure diffusion being steeper than the CO2 gradient, equivalent measures of gases are traded. Figure 7.24 Partial pressure gradients in the body. Promoting gas exchange. Top. Oxygen and carbon dioxide gradient exchange through respiratory membrane. Bottom. Gradients across systemic capillary membranes. 2. Ventilation, perfusion, coupling ventilation. Amount of gas reaching the alveoli and perfusion blood flow in pulmonary capillaries is needed for efficient gas. Ventilation is often poor in alveoli, thus the oxygen pressure is low in that area. Along these lines, the terminal arterioles narrow down, diverting the blood to areas where PO2 is high, and oxygen pickup turns out to be increasingly productive. When ventilation is most extreme in the alveoli, pulmonary arterioles expand, boosting blood flow into the associated pulmonary capillaries. 3. Structural characteristics of the respiratory layer. A change in partial pressure of O2 PO2 in alveoli affects the diameter of arterioles, while a change in partial pressure of CO2 PCO2 in alveoli leads to fluctuations in the diameters of the bronchioles. Such changes in the diameter cause the alveolar ventilation and pulmonary perfusion to get synchronized. The pulmonary arterioles constrict, making the airways dilate, which helps in bringing blood and airflow to a closer physiological match. The presence of high PO2 and low PCO2 in the alveoli causes bronchioles to compress, which forces the blood to flow into the pulmonary capillaries. Although such changes take place for adequate gas exchange, complete balance of ventilation and perfusion does not take place due to other factors, such as gravity leading to disparities in flow of blood and air as well as mucus present in the alveolar duct causing unventilated areas. In addition to this, the shifting of blood from bronchial veins 104 milliliters Hg in alveolar air to pulmonary venous blood 100 milliliters Hg is shown in figure 7.24. 4. Surface area and thickness of the respiratory layer. In normal lungs, the thickness of the respiratory layer is nearly 1 micrometer. Large surface area would allow more gas to diffuse across the membrane. The surface area for gas exchange in alveolar sacs is nearly 90 meters too, almost 40x higher than the surface area of the skin. Internal respiration. The opposite takes place during internal respiration than for external respiration. Tissue cells constantly use O2 and give out CO2. Since PO2 in tissues is lesser 40 milliliters Hg in contrast. To systemic arterial blood 100 milliliters Hg, O2 from the blood travels into the tissues until equilibrium is achieved. Simultaneously, CO2 travels into the blood. Gas transport by blood. Blood is primarily involved in the carriers of O2 and CO2 between internal and external respiratory sites. Transport of oxygen. Transportation of O2 in the blood takes place in two fashions. It binds to hemoglobin and gets dissolved itself in plasma. The solubility of O2 in water is weak, of which 2% is in the dissolved form. Since this value is needed to provide oxygen in the body, hemoglobin solves the issue, rests 98%. Association of oxygen and hemoglobin for transportation to take place. Oxygen combines with hemoglobin forming oxyhemoglobin HbO2 with the help of iron-containing heme group present in its 4-polypeptide chain. Hemoglobin then releases oxygen referred to as deoxyhemoglobin HHV. The reversible loading and unloading of O2 is equated as HHV plus O2 rightwards harpoon over leftwards harpoon HbO2 plus H plus. The HV changes shape when the first oxygen binds to iron. Loading and unloading of single oxygen enhance the loading and unloading of the next, and the chain goes on. Eventually, uptake of 3 more O2 molecules is facilitated. Once all 4 heme groups are bound to oxygen, the hemoglobin gets fully saturated, otherwise it is partially saturated. The affinity of HV for O2 depends on the oxygen saturation. Influence of PO2 on hemoglobin saturation. The relation between PO2 of blood and hemoglobin saturation is not linear since affinity of hemoglobin changes with O2 binding, displayed in oxygen hemoglobin dissociation curve in figure 7.25. The curved S shape has an abrupt slope for PO2 values 10 to 50 milliliters Hg, which flattens out 70 to 100 milliliters Hg. During normal resting conditions, arterial blood HV is 98% saturated. PO2 equals 100 milliliters Hg, while 100 milliliters of systemic arterial blood holds around 20 milliliters of oxygen. When arterial blood passes through the systemic capillaries, about 5% of 100 milliliters O2 is released, yielding a 75% saturation rate for HV and 15 volume percent of O2 in venous blood. Figure 7.25 Oxygen hemoglobin dissociation curve HV saturation changes with blood PO2. Note that HV is almost saturated at a PO2 of 70 milliliters Hg. loading and unloading of O2 in the steep portion of the S curve. Nearly 25% of HB-bound O2 is unloaded to resting tissues in systemic circuit. Thus, the rest 75% is still saturated with oxygen. Exercise causes additional 50% unloading, leaving only 25% of hemoglobin saturated. PO2 dropped to 15 millimeters Hg. The oxygen-hemoglobin dissociation curve reveals two other important facts. First, hemoglobin saturation takes place at PO2 of 70 millimeters Hg, and additional increments in PO2 yields minimal increases in oxygen binding. The reason behind this adaptive value is that O2 loading and delivery to the tissues is adequate even if the PO2 is below its normal level. Lastly, since O2 unloading takes place on the steep portion of the graph curve, even a small descent in PO2 leads to a substantial rise in unloading. Frequently, nearly 25% of bounded oxygen is unloaded throughout a single systemic circuit, while sufficient volumes of O2 are yet accessible in venous blood. Consequently, if O2 dips to deficient levels, more O2 will dissociate from hemoglobin with no increase in cardiac output. Influence of other factors on hemoglobin. Other factors, such as temperature, blood pH and concentration, PO2 and PCO2 also affects the loading and unloading of oxygen from hemoglobin. The blood concentration of a chemical called 2,3-phosphoglycerate BPG binds reversibly with hemoglobin and is produced by red blood cells RBCs with help of glycolysis. An increment in temperature. PCO2, H+, or BPG levels in the blood reduces Hb's binding strength for O2, thereby improving O2 unloading. Conversely, a reduction in any of these factors enhances Hb's binding strength for oxygen. In systemic capillaries, the oxygen unloading is the highest gain, thus all these factors are at its highest peak there. Transport of carbon dioxide. Usually, cells produce nearly 200 milliliters of CO2 per minute. Blood carries carbon dioxide from cells to lungs in three ways. 1. Dissolved in plasma, 7-10%. Nearly 7%-10% of CO2 is transported within plasma in dissolved state. 2. Chemically bound to hemoglobin, over 20%, like oxygen. Dissolved CO2 is bound to hemoglobin as carbaminohemoglobin. Carbaminohemoglobin binds directly to amino acids of globin and hemoglobin, thus does not interfere with oxyhemoglobin, which is bound to heme. PCO2 and the Hb oxygenation influence the carbon dioxide loading and unloading. CO2 quickly dissociates from Hb in the lungs, where the PCO2 is and is loaded in tissues where PCO2 is higher comparatively. 3. As bicarbonate ion in plasma, about 70%, majority of CO2 enters the plasma as bicarbonate ions, HCO3. Once dissolved, CO2 diffuses into the RBCs. It combines with H2O, forming carbonic acid H2-CAM3, figure 7.26. Since H2-CAM3 is not stable, it separates into hydrogen ions and bicarbonate ions is equated here. CO2 plus H2O rightwards, harpoon over leftwards, harpoon H2-CAM3 rightwards, harpoon over leftwards, harpoon H plus HCO. This reaction, although occurring in plasma, is 1000x faster in RBCs as they contain carbonic anhydrase, an enzyme which reverses the reaction to carbonic acid. Hydrogen ions release bind to hemoglobin, triggering the Bohr effect in a way that CO2 loading improves O2 release. The released hydrogen ions change the pH 7.4 to 7.34, making it acidic. HCO3, on the other hand, transports from the RBCs into the plasma. To counterbalance the rush of these ions from red blood cells, chloride ions Cl move from the plasma into the RBCs, known as the chloride shift. This process is reversed in the lungs, figure 7.26. When blood passes through pulmonary capillaries, PCO2 drops 45 millimeters Hg to 40 millimeters Hg. For this to happen, HCO3 re-enters the RBCs and binds with hydrogen ions to form carbonic acid. Carbonic anhydrase then splits it to release CO2 and H2O. This carbon dioxide plus the CO2 released from Hb, along with the one diffused in plasma, moves from the blood to the alveoli. In external respiration, oxygen diffuses across the respiratory membrane from the alveolus to the capillary, whereas carbon dioxide diffuses out of the capillary into the alveolus. Figure 7.26 transport and exchange of CAM2 and O2, external respiration. The Haldane effect. The oxygenation of the blood markedly affects the CO2 transportation. The property called the Haldane effect explains how oxyhemoglobin forms carbaminohemoglobin. The entry of CO2 reduces the oxygen's affinity to associate with Hb, Bohr effect. This allows more CO2 to combine with hemoglobin, Haldane effect, which encourages CO2 exchange. Control and regulation of respiration. The primary respiratory rhythms are mostly controlled by brain centers, chemoreceptors, and other reflexes in the brainstem. Neural mechanisms. The respiration is majorly controlled by neurons in the medulla and pons region. 1. Medullary respiratory centers. Number of neurons are found in the dorsal respiratory group (VRG) and the ventral respiratory group (VRG) within the medulla oblongata. The VRG is an integrated and rhythm-generating center which holds clubs of neurons that fire during inspiration and expiration with mutual inhibition. During the fire of VRG's inspiratory neurons, a rush of impulses stimulates the diaphragm, which causes thorax to expand and gases to flow in. The reverse takes place when expiratory neurons fire, causing the lungs to recoil. This cyclic on-off motion happens continuously and produces a respiratory rate and rhythm known as eupnea. A rate of 12 to 15 breaths each minute, 2's inspiration, 3's expiration, is generated. This standard respiratory rate and rhythm are referred to as eupnea. Breathing stops completely once a certain mixture of VRG neurons is entirely repressed due to excessive morphine or alcohol. 2. Pontine respiratory centers. The pontine respiratory center controls and modifies the activity of the medullary respiratory center by easing out the cyclic on-off motion mentioned previously. Human physiology is a branch of science that deals with the study of human cells, the organs formed by these cells, and their organization to support the various functions of the body in health and disease. It focuses on the body's ability to maintain a steady state of homeostasis. Homeostasis is defined as a state of a stable internal environment maintained by living organisms through regulating various physiological processes while adjusting to external conditions. In this branch of science, human response to internal and external stimuli such as changing environmental conditions, changes in activity levels, and disease conditions is measured and is utilized for the betterment of human health. Levels of organization. The universe is made up of matter, defined as any substance that occupies space and has mass. Matter consists of one or more pure, unique constituents known as elements, i.e., hydrogen, oxygen, carbon, nitrogen, calcium, and iron. Atom is the smallest basic unit of these elements. Each atom is further divided into subatomic particles known as proton, electron, and neutron. Atoms combine to form molecules found in living organisms, such as proteins, sugars, and water molecules. Molecules go on to form the building blocks of the body's structure. The cell is the smallest, most basic, independent unit of life, which contains all the structures of the human anatomy along with different functions that take place in the human body. All the various functions are commenced by the cells. Each cell in the body is made of flexible membranes that surround the cytoplasm, a water-based, fluid-containing structure in which the operative units of cells called the organelles are suspended. Figure 7.1. Cells of similar structure and function group to form tissues and group of different tissues assemble to perform a similar function, forming an organ. Each organ is part of one or more physiological functions. A group of organs which perform similar functions make up the organ system that is important to carry out the physiological needs of the body. Figure 7.1. A typical human cell. A prototypical human cell comprises of internal organelles in the cytoplasm and inside the nucleus. The human body cell is made of extremely organized structures called as intracellular organelles, which contribute to cellular functions. Majority of organelles are protected by membranes consisting of lipids and proteins, which include the plasma membrane, nuclear membrane, lysosomal membrane, endoplasmic reticulum membrane, mitochondrial membrane, and Golgi apparatus membrane. Tissue types. Tissues are a group of cells with a similar structure that performs a specific function in the body. Tissues are classified into four basic types: epithelial tissue, connective tissue, muscle tissue, and nerve tissue. Figure 7.2. 1. Epithelial tissue, also known as the epithelium. It forms the covering of body surfaces, internal organs, and lines of body cavities. It also forms the lining of some passages and some glands. 2. Connective tissue. As the name suggests, the cells of connective tissue connect the cells and organs of the body together. Some of the important functions of connective tissue include support, protection, and integration of different parts of the body. 3. Muscle tissue. This tissue consists of cells specialized for contraction to bring about animal sciences. Movement. The cells of muscle tissue possess the ability to excite and respond to stimulus. 3 different types of muscle tissue are skeletal muscle, also known as voluntary, smooth muscle, and cardiac muscle present in the heart. 4. Nerve tissue, found in the nervous system. Nerve tissue consists of specialized cells that can generate and transmit electrochemical signals in the form of nerve impulses. They possess an excitable characteristic that links different regions of the body and assists in the maintenance of homeostasis. Tissues are classified as epithelial tissue, forms covering the outermost surfaces of the body. Connective tissue, binds cells and organs of the body. Muscle tissue, excites figure 7.2 different types of tissue. And responds to stimulus and contracts to provide movement. And nervous tissue connects between different parts of the body by propagation of electrochemical signals. 5. Epithelial tissue. Cells of epithelial tissue form a lining and covering of the outer surface of the body. The cells are organized with very little or no extracellular spaces present between them, attached to one another by means of desmosomes. The tight organization of cells renders epithelial tissue with the property of protection. They are anchored to a connective tissue which provides the cells with nourishment as the epithelium lacks blood vessels. The epithelial tissue is classified into two types. 1. Lining and covering epithelium. 2. Glandular epithelium. Lining and covering epithelium. As the name suggests, this class of tissue form the outer covering of skin, respiratory tract, digestive tract, reproductive tract, and urinary tract. Blood vessels, serous membranes, and other hollow internal organs are lined by epithelial tissue. The cells are organized in the form of a single layer or several layers. Lining and covering epithelium are further classified based on the shape and arrangement of cells into layers as big. 7.3. Simple squamous epithelium. The word simple refers to a single layer of cells that rest on the basal surface and give the appearance of thin scales. The cells are flattened, horizontal, and elliptical in shape with a centrally located nucleus. This epithelium assists in the exchange of nutrients, waste and gases, and protection. It is found in areas where diffusion or filtration occurs. Example, lining of the lymphatic system, alveoli of the lungs, and glomerular capsule. Simple squamous epithelium lining heart and blood vessels are known as endothelium. Similarly, simple squamous epithelium lining the outer surface layer of the serous membrane, such as peritoneum, is known as mesothelium. Simple cuboidal epithelium. Single layer of cuboidal-shaped cells that have a centrally located round nucleus. This type of tissue is found in areas where absorption or secretion occurs. Example, forms a lining of kidney tubules, surface of ovaries, ducts of pancreas and thyroid gland, and anterior surface of capsule of lens of the eye. Simple columnar epithelium. Single layer of elongated cells that are column-like with a nucleus located in the basal end of the cells mainly for the purpose of protection, secretion, and absorption. Example, lining of small intestine and uterus. Simple columnar epithelium can further be classified into ciliated and non-ciliated simple columnar epithelium. Ciliated columnar epithelium has additional finger-like projections called microvilli on their apical surfaces for increasing the surface area of absorption. The ciliated columnar epithelium is found in the lining of fallopian tubes and parts of the respiratory system, where the beating of the microvilli helps remove particulate matter. Non-ciliated simple columnar epithelium does not have uniform microvilli and helps in secretion and absorption. Pseudostratified columnar epithelium. Pseudo means false and stratified means multiple. Pseudostratified columnar epithelium tissue is a single layer of cells that appears in multiple layers, stratified due to the presence of multi-layered nuclei. The nuclei are located at different levels instead of grouping at the base, thereby giving the look of various layers. However, all the cells rest on the basal lamina. The function of this epithelium is the secretion of movement of mucus. The cells have hair-like projections on the membrane known as cilia that aid in the removal of bacteria or foreign particles by trapping. Example, lining of the airways of upper respiratory tract, and movement of egg in the fallopian tube. Stratified columnar epithelium. It present in areas subjected to wear and tear. It protects the underlying tissue as it is present in many layers. It can be further classified as stratified squamous epithelium. In this tissue, the apical layer consists of squamous cells, whereas the basal layer consists of cuboidal and columnar cells that become flattened as they move upwards. The major role is the protection of underlying tissues. Example, lining of the vagina, oral cavity, the throat forms the epidermis, and the top layer of skin. Stratified cuboidal epithelium contains 2-3 layers of cuboidal-shaped cells present in the apical layer. These cells aid in protection, secretion, and absorption. Example, the lining of sweat glands, salivary glands, mammary glands, and pancreas. Stratified columnar epithelium consists of 2 or more layers of irregularly shaped cells with apical layer having columnar cells. The function is protection and secretion. Example, lining of the urethra, pharynx, esophagus, and stomach. Transitional epithelium. In the apical layer, there are various types of cells which consist of cube-shaped, on-relaxing, and elongated, on-stretching. The function is protection and dispensability. Example, inner lining of urinary bladder and lining of ureters and urethra. Figure 7.3 types of lining and covering epithelium. Simple epithelial tissue is organized in the form of a single layer of cells where all the cells rest on basement membrane, and stratified epithelial tissue is organized as 2 or more layers of cells where only basal layer rests on the basement membrane. Glandular epithelium forms the lining of various glands of the body, such as pituitary glands, adrenal glands, etc. A gland is a specialized structure that has cells epithelial to produce and secrete specific substances chemical into body fluids or ducts. These chemical substances, which is fluid-like, are called secretions and contain proteins. Glandular cells obtain substances from blood and chemically change them by an active process into products called secretion, which are then released from the cells. Based on the secretion of products, glands are classified as endocrine and exocrine. Glands can also be classified based on the number of cells present in them as unicellular and multicellular. Endocrine glands. These glands secrete their products, called as hormones or chemical messengers, directly into the blood or body fluids. All hormonal glands are endocrine glands. The products of endocrine glands, which are released into the interstitial fluid, are diffused into the bloodstream by exocytosis. From bloodstream, the hormones are then delivered to the target cell. Hence, endocrine glands are also known as ductless glands. Endocrine system is crucial for the integration and regulation of bodily responses. Example, hormones produced by intestinal cells cause pancreas to release enzymes that help in digestion. Exocrine glands. The exocrine glands release their products and secretions through ducts that secrete the products into an open space. Examples: sweat glands, oil glands, salivary glands, and tear glands. Based on the number of cells present, exocrine glands can be classified as unicellular and multicellular. Unicellular exocrine glands. These glands release their secretions directly by exocytosis. Unicellular exocrine glands are found within the epithelial lining of intestinal and respiratory tracts. Mucin, a glycoprotein, is produced by these glands, which when dissolved in water, form mucus for protecting and lubricating surfaces. Multicellular exocrine glands. The secretions of multicellular exocrine glands are released on the epithelial surface. Multicellular exocrine glands have more than one cellular unit, an epithelium-derived duct and a secretory unit. The secretory unit is surrounded by connective tissue, which supplies blood vessels and nerves to it. Multicellular exocrine glands are further classified based on the structure of their duct as 1. Simple glands, contains an unbranched duct. 2. Compound glands, contains a branched duct. Compound glands on the basis of secretory units are further classified as tubular, alveolar, and tubulovular. Tubular compound glands have secretory cells that form tubes. In case of alveolar, secretory cells form small sacs. Similarly, tubulovular have both tubular and alveolar secretory cells. Figure 7.4. Modes of secretion. Based on the mode of secretion, multicellular exocrine glands are classified as merocrine glands. The secretory cells of merocrine glands excrete their products by exocytosis without causing any damage to the secretory cells. Salivary glands, pancreatic glands, and nephrin glands are examples of merocrine glands. Apocrine glands. Secretion occurs in such a way that membrane-bound cytoplasm from the apical surface of the cells pinches off into the lumen of the duct. Examples include mammary glands and axillary sweat glands. Holocrine glands. Secretion consists of secretory product along with its own disintegrated secretory cell. The plasma membrane ruptures when the product is released. Example is sebaceous gland present in skin. Figure 7.4 types of multicellular exocrine glands. The multicellular exocrine glands may have simple or branched ducts with secretory units. Connective tissue. Connective tissue is one of the 4 main types of tissues in the human body. Connective tissue provides structural and metabolic support and is the most abundantly distributed tissue. It serves as a connecting link for binding, strengthening, and supporting all the other body tissues. The main components of connective tissues are ground substance, fibers, and cells. The group substance is clear, viscous, colorless, and fills space between cells. Ground substances constitute the fluid extracellular matrix. Protein fibers constitute the solid extracellular matrix, and together they make up the matrix. The matrix secreted by the connective tissue determines tissue volume and specialized function. Connective tissue is broadly classified into 2 types: embryonic connective tissue. Meet mature connective tissue. Embryonic connective tissue. 1. Mesenchyme. The mesodermal germ layer of the embryo is the source from which all the connective tissue is derived. Mesenchyme is the very first connective tissue that is developed in the embryo, and in the later stages, it is a stem cell line from where all the other connective tissues originate. In adult form, mesenchymal cells are present in irregular, scattered patterns and play a role in the repair and replacement of connective tissue after injury or damage. Example: present under skin in embryo along the developing bones, whereas in adults, along blood vessels. 2. Mucous connective tissue. It is also known as Warden's jelly, consisting mainly of ground substance hyaluronic acid and chondroitin sulfate. It also has low amounts of collagen and reticular fibers. Example: found in umbilical cord. Mature connective tissue. 1. Loose connective tissue. Loose connective tissue helps the surrounding neighboring cells and tissues by allowing the diffusion of water, salts, and various nutrients. It is also present in many tissues, where it prevents shock absorption. 2. A relar connective tissue. It is the least specialized of the various types of connective tissues. In addition to collagen, elastic, and reticular fibers, it consists of different types of cells: fibroblasts, adipocytes, mast cells, and macrophages. It has an open framework with a major role of filling spaces muscle fibers surrounding lymph and blood vessels, and also provides support, strength, and elasticity to the organs in the abdominal cavity. A real connective tissue also holds blood vessels and capillary beds. Example: the subcutaneous layer of skin.